So welcome everyone to this symposium. It's from the neuroscience track of the Mind Foundation, and it's about addressing the challenges and the opportunities both that we have in bridging the gap between basic science and clinical research in psychedelic neuroscience. We have 5 speakers. Each one will give a talk first, and at the end we have a 30-minute panel discussion where I'll be happy to take all other audience questions as well as try to bring these speakers together. We have today first Professor Dietmar Schmitz, then Professor Robert Malenka, Dr. Torben, Professor Kim Kuypers, and Professor Olson. Professor Olson will join us online. He is in US and he couldn't be here physically, so he will give an online talk. And yeah, it's very early in the West Coast, but that's why we have him at the end, and he will also join us for the panel discussion. So I'm looking forward. And first I would like to invite Dietmar Schmitz, who is also my supervisor at the Charité. So Dietmar, the floor is yours.
Yeah, thanks, Prateep, not only for the start, the introduction, but also for the initiative of triggering this event, for the coordination, and for the chance, the opportunity to discuss with you some of our ideas and also some of our data. I decided to have a kind of a jump start. Could you show perhaps these slides? I prepared a few. I was trying to. It's a tile slide, which I would like to skip. Can I use then the remote control here? Is this something? Sure. This is another one. This is Torben. My name is Dietmar Schmitz. I'm the first speaker. It's not connected. Then I perhaps have a kind of a spontaneous few sentences. So I had a long-standing interest in, not directly into psychedelics initially, but into serotonin, which is a transmitter, or some people call it neuromodulator, and how this affects the brain. I actually started working on this, and now I realize I'm a bit older. This was more than 30 years ago, 31 exactly. This is also not me. Dietmar Schmitz, but I mean, this would be actually interesting. I mean, I could try to give your talk and then we switch a bit. Might be a bit more creative. This looks like the one. And we are working in different brain areas, and I will come to the kind of cellular effects and then the network effects of serotonin, which is a kind of the intrinsic ligand for all these, or many of these psychedelics. Here we go. Who am I? Here. And I will start with an anecdote. It's a kind of a personal encounter into this area. So I came from the west coast of the US, from Berkeley, San Francisco here to Berlin in 2002, roughly. And the president of the Humboldt University invited me to have a chat, and he's a physicist, a great physicist, and he said, well, Dietmar, we should really trigger interest in young people and students for our stuff we are doing. Natural sciences, all people do law school, and it's also important, of course, but it would be great to have natural science people, or medicine, psychology. And he asked me to coordinate with him and others a kind of a university for, this might be too close here, for kids, children. And this is a kind of a general thing I like to do. The perspective of kids on certain things. So I had a huge meeting, so 700 kids in the audience, parents, was in the kind of a cinema, plus teachers. And it was about the brain, and you know, this was primary school, so you had really young kids, 5 years old. They asked more about where's the brain located. And then older kids, 12, 13, they Googled me and they found out that I had just a paper published on cannabinoids, on cannabis, how this would affect the brain, endocannabinoids, and they asked me then how cannabis would affect the brain. So kind of broad range. I had first to synchronize all these kids, very noisy, and then I did a massive neurosurgery here. All of our kids here, and there was indeed a brain located, actually a YouTube thing, but it was fun. But what was so rewarding then, so I mean, this sounds perhaps a bit full of ourselves and arrogant, but we had lots of invitations afterwards, and we really introduced something also into schools. Yeah, it's not only me, but also other people going into schools. And one thing I received from a teacher from also here from Germany, from Münster, was an essay, 4th class, young girl. And it's in German, but I translated it in English. And yeah, it's perhaps my personal thing. I can laugh over and over again. Perhaps this triggers also your serotonin release. Let's guide me through this. With the brain, we think that we think. I mean, this is philosophical and also not so easy to prove. Yeah, we think that we think. We also need it for headaches. It's located in the head, right behind the nose. When we sneeze, it drips. And now the two last lines is best. The brain is a very sensitive organ. That's why most people use it very rarely. So I think it's great, and perhaps I should stop here, so hopefully the questions are then a bit easier. So now I also try to do science. And this is some people, I mean, there are all these things for all these molecules, the God molecules, the mood molecule, it's called sometimes the magic. I call it 5-hydroxytryptamine. That's a chemical form. It sounds a bit boring, but that's serotonin. I started working on it, as I said, in the beginning of the 90s. So there it was clear where it's roughly located. I have also a slide on this. So here's roughly the structures of those. And you see that these, I mean, it's a bit academic, the terminology, what is psychedelic, what's not psychedelic drugs, classical, non-classical. But you see the structure is, for some of them, the DMT, the psilocybin, and also the LSD, based on this structure here, which is an intrinsic ligand. And I thought I'd give you roughly a short introduction into serotonin, because me as the first speaker, and then Rob can directly jump into the real science. And I think Rob, he had also a slide on where serotonergic neurons are located. I have a slightly different one here. It's here, so we know in the mammalian brain we have serotonergic neurons in the so-called DRN, dorsal raphe nucleus. And perhaps not so easy to see here, but they project all over the place. These arrows here are thought to be axons, projecting into many different brain areas. And if you now zoom into one target region here, somewhere, let's say in the prefrontal cortex, but doesn't matter, it's kind of the general principle, you can look at this synapses. I am not sure about the background or all of you in terms of sciences, neuroscience, so I thought I give a very broad initial introduction. So you have this axon. At the end of the axon, you have the terminal here with the transmitter, the synaptic cleft, which was a discussion for quite some time in neurosciences, for about 100 years, whether everything is directly connected or whether there are these gaps, units. And then you have these kind of proteins here, which are the receptors. And you can modulate the serotonergic system. And the classical way people do in the clinics right now is blocking these uptake of via the transporter here. And there you can slowly and slightly increase the serotonergic level, the serotonin level. You can do also a much stronger treatment, fast and strong. And I think Rob will talk about this. Mainly it's about MDMA. And you get this massive increase here in serotonin. And last but not least, what you also can do is you can bypass this and directly activate these receptors here. This is shown here, for example, for a classical, one of the serotonin receptors, 5-HT2A. And then you can also have, of course, dependent also on the concentration, increase the serotonergic or enhance the serotonergic system. And when I started, the neurons were localized, and the first cellular effects have been described on neurons of serotonin. Actually, my previous supervisor was early on in the 80s working on this. But the receptors were not really cloned. And then all these receptors, were cloned one after the other. And I have to say, I was a bit nervous. I mean, most transmitter systems, they have then 1, 2, 3 receptors, but here are many. And they have even subclasses, 5-HT1A, 1B, 2A, 2B, 2C, and so many of those. I never gave up completely, and you will see I brought a small story with me today. We are still working and busy with, but it's a rather complex system, and I think the good news is we all have to do a lot of work. And this is now the story. So what we thought is, so how is serotonin affecting the brain? The synapse, this is something we worked on a lot. It can modulate the synaptic transmission. It can directly affect single neurons. And it can affect, and the question was also for us, will it really affect also the overall network of the brain? And what we did is doesn't matter, the resolution for whatever reason is a bit down here. But what we did is we started to record in a brain area, which I would like to briefly introduce to you, which is the so-called medial entorhinal cortex. This was actually the second part of my PhD. At that time, it was very little investigated. So people thought, oh yeah, that's simply an input into the so-called hippocampus, which is the most, I think, investigated brain area, perhaps. And we started to characterize different cell types, and not because of me, because of others. It's a quite well-known area. Actually, 2 Nobel Prizes were given now for the discovery of so-called grid cells, which are quite specific for the entorhinal cortex or the neighbor structures. It's located here, very deep in the brain. It's not so easy sometimes to target. And what was also for us interested, it receives lots of serotonergic inputs from the nucleus I was talking about into the, it's a heavy input into the entorhinal cortex, and there are also tons of receptors. And what I also found exciting is it's a kind of, some people would call it the multimodal center. We receive tons of this inputs and outputs into this entorhinal cortex area. And what it does under baseline conditions in head-fixed anesthetized animals, you will find a kind of a brain rhythm. Some people call it the default mode, a kind of an intrinsic mode. We're working a lot on oscillations, and it's still not clear how this oscillation is coming about. But what we wanted to do is to study how serotonin affects this baseline brain rhythm. And we use different methods, and I will guide you through. So we are physiologists, so we do the so-called patch clamp technology, single cell. We try to do in vivo recording, pharmacology, genetics, optogenetics, and we use genetically modified animals. And this is the first recording session we did is with an electrode, which is called silicon probes. This is shown here, and we introduced several of those into the brain, here into the medial entorhinal cortex. To be honest, initially we sometimes failed and didn't get it in the right position. And then you can record for many neurons at the same time. And these are these action potentials, the spikes of the cells. And what we did then, we applied after kind of a baseline situation, this is shown here for the up and down states, and for the spikes, then MDMA. And I have to say, I mean, sometimes we work on these kind of small effects, 10, 20, 30%, but this was, okay. But this was really massive, I have to say. I've rarely seen such an effect. It's completely wiped out. It's gone. These up and down states, so this is true here for the up and down states, and also these spikes we were measuring. And then after washout of the MDMA, it's slowly recovered. So the animal actually and the cells are still alive. It's good to have a recovery. And here we plot a kind of the time course for this effect for MDMA. So you have a baseline, which is usually without MDMA, quite stable. This we also, it's a kind of an intrinsic control because we were using anesthetics to be sure that there is not a kind of a spontaneous rundown. And as you can see here, MDMA, as shown before, it has this massive effect both on the up states and also on the spikes. And then we did an additional experiment because MDMA is not only releasing serotonin, it will also release oxytocin, it will also release dopamine to a lesser extent, but who knows, yeah? And we used another substance which is called fenfluramine, FFA. And as you can see here, this FFA, here in kind of yellow, orange, it's more or less mimicking this effect. It's also completely wiping out here this activity in the entorhinal cortex. So we are mechanistic people, so we thought, well, this is a nice starting point. So now we have a big result here, a strong result, strong effect. Let's look into the underlying cellular or network mechanisms. And these in vivo situations are, on one hand, it's a kind of the real world, so to say, but on the other hand, it's technically sometimes a bit challenging. So one method we used in the initial part of the study was, it's called spike sorting. I don't want to go too deep into it. It's also not perfect, but you can get roughly in terms of the spike shape here, whether it's broader spikes, narrower spike, this so-called repolarization, you can get an idea what kind of cells might be involved. And we saw that these cells which broader spikes, putative, excitatory cells, principal cells, projection neurons, you might call them, were mainly not affected by MDMA. Then there were cells which have a very narrow spike. They are so-called GABAergic interneurons or subclasses of GABAergic interneurons. Some, they are named basket cells or path basket cells. They have very fast spikes, 5 times faster than the others. But there was kind of an intermediate group. They were not broad, they were not very narrow, and those were the cells which massively increased with MDMA. So we thought, okay, what's going on there? What could this be? Again, this quality is not great, but I think I can guide you through here. So what we did then next is to get a more handle on what might be the underlying mechanism. We switched into another method, in vitro electrophysiology. What you can do is you can cut brain slices, and then it's easier to do cellular recordings, to do identified recordings, to do pharmacology, and so on. And first, and thanks to Prateep, we had another study going on, which was the requirement actually to do this. We had to establish these kind of brain rhythms, or to be able to measure these brain rhythms in vitro. And what we did, we recorded with the patch clamp technology from several cells at the same time. Here we have setups where we can do up to 8 cells at the same time. Here's an example with four such cells. And you see there's activity in all these 4 cells, spikes, subthreshold activity, and this is highly synchronized if you do this kind of analysis. And if you now in vitro apply MDMA or serotonin, so a kind of a mimicry, so the ligand, which the 5-hydroxytryptamine, which we did here at relatively low concentrations, and on all the slices and cells we investigated, you see this massive effect of serotonin. Here it's a bit delayed, but it's also going down, and here you see all these individual recordings, and yeah, here's a summary plot. Again, in vitro, I guess we can mimic the situation from in vivo with serotonin. It's kind of reassuring. You get this massive reduction. Then we thought, okay, which receptors might be involved, which is something I sometimes don't like to do because the pharmacology is a bit tricky. So many drugs are out there, and but we were, I would say, partly lucky. We started with 5-HT1A, there was no effect, 5-HT3, no effect, but 5-HT2A agonists, so also the receptor where usually psilocybin or psilocybin or LSD would bind to, not exclusively, but mainly. And we used another drug here, it's called alpha-methyl 5-HT. It's a relative selective 5-HT2A agonist. And you can see we have less ends here, recordings, but it mimics the effect of serotonin. So it looks like that at least 5-HT2A is involved. And we had also done some antagonist experiments with ketanserin. And to be honest with you, it was reduced. The effect was clearly reduced. I have also one slide on this, but it was not completely gone. I mean, it sometimes might be other receptors, might be also that it's a matter of concentration. And now coming back to the the cell types being involved into this effect, and I will cut this a bit short. So what we initially did, because the entorhinal cortex is not fully characterized as other brain regions, there are tons of different cells which we still don't understand fully. We had a kind of a massive brute force recording for many cells, and again, in vitro, we had the result that these kind of medium board spikes, these cells are sensitive to serotonin. And then what we did is we used a kind of a transgenic approach. We had animals, many different types of animals, path interneurons, but also so-called somatostatin. It's a subtype for interneurons, quiline, and we brought in first a reporter. It's called here TD tomato, so you can identify these cells here in the entorhinal cortex. And then what you can do is you can do this kind of targeted patching. So here's a slice, looks like a slice of bread here actually with a biorender, and you patch these cells here from the transgenic animal. And if you do so, we can characterize the cells. So it looks like GABAergic interneurons. And as you can see here with serotonin, you get a clear depolarization of the cells, which is not the case for the principal cells. There you have a slight hyperpolarization. So a clear, strong effect. And this effect is antagonized by ketanserin, the 5-HT2A receptor antagonist. So then next, what we wanted to make sure is, I mean, one could call it control experiment, but I think it's a bit more than a control experiment. We used an optogenetic approach to make really sure that the somatostatin interneurons, which initially was a bit hard for us to believe that they are really so effective and so strong there, because the focus in research was so heavy on other interneurons, we used again a kind of a genetic approach, an optogenetic approach. So we used these mice and we brought a light-sensitive channel into these cells. This is something a great biophysicist here from Berlin discovered, and somebody from Stanford. They both did great work on this technology. It's very helpful in system neurosciences. So we brought this into these cells, and now I have to guide you through this slide. So we record here from cells, sometimes several cells at the same time, and then you get these kind of up and down states. And now you switch on the light, and whoop, it's more or less gone, the activity. And then turning off the light, the activity comes back. And this you can also then nicely show here in this raster plot for spikes and up and down states. Then I think I run out of time, so second last slide. We also thought about the other way around. So we used a kind of an antagonistic effect, not pharmacologically, but so-called chemogenetically. This is more for the experts perhaps here. We did a kind of a crossbreeding with two lines. Actually, this was homemade and actually also done in our lab. Crossbreeding with with the interneuron here. And what you can do then is we first had the effects of serotonin, and then you can activate these receptors. I mean, it's very specific, but what you can see here is that you can bring the activities then back. So the idea being that first serotonin would affect, perhaps depolarize these interneurons, and now you take this back, not to baseline, didn't come back completely, but it was a clear effect, I would say. We did also the other way around, first the chemogenetics, then serotonin. This, I would say, was quite clear. All right, I think I have to come to an end here, but I want to thank the people. So Prateep, I mentioned, he was the one who's setting up really the up and down states recordings in vitro. He was also the one training Roberto, a PhD student in our lab, and he did the MDMA study. And let's also finish with another anecdote. My last 2 minutes. I started with kids, and I also will conclude and finish. Here, these drawings here from my kids, Hannah and Lena, and they were 5 and 7 years old. And these are the so-called hippocampus. So these seahorses, which is the brain area we are mainly working in. And they did these drawings, which often on seminar, lecture series, we use this as a background, which is nice. But the story goes like this. So I was at home and putting some slides together, and was some computational work. I was on my way to Jerusalem, Hebrew University, and they have very strong mathematicians there. And I was a bit nervous, I have to say. They can give you really a tough time. And said, what are you working on? She was 5 years old. And I said, oh yeah, this is the brain, a bit of disease. Actually, there was nothing on disease, actually. But, and okay, talk was mediocre. And few months later, again, I was doing a few things at home, and I was on my way to Zurich. And again, Hannah was there. It was a Sunday. Oh, what are you doing? And to be honest, there was not much modification. One data slide left and right, and one additional perhaps. And what are you working on? I said, well, it's about the brain and disease. And she looked at me, mouth open, said, what? You are still busy with the brain? And I mean, for me, it was a kind of an eye-opener. She was, of course, very frustrated with her dad. She thought, oh my God, this guy is so slow and dumb. Why is he not working in astrophysics? And but for me, it was clear. So we do little things, at least we do little things, but as a kind of a collaborative community, I think we can do more, and some people are a bit faster than I am. Okay, so thanks so much for your attention.
Yeah, we can take 1 or 2 questions.
Okay.
So questions right now. We can also have the questions later if there's no burning questions immediately. Any questions? 1 question?
What do you think? What do you think that made your result? In astrophysics.
Yeah. I mean, I mean, for us, as I said in the beginning, for us is the investigation of serotonin on neurons. How is serotonin? It's a kind of a really basic research. How would they affect neurons, the synapses circuit, and so on. I can't tell you whether it has anything to do with behavior. Well, this is something we would still love to do. And I also can't tell you that whether psychedelics or MDMA would cause some of their effects via the entorhinal cortex. So still work in progress, so I can't say. But it's, I would say, a starting point. We have a big effect.
Thanks, Dietmar, very much. So now we move on to the next speaker, Professor Robert Malenka. He is here all the way from Stanford, and he will talk about mechanisms of the prosocial actions of MDMA. Thanks, Rob, for coming.
Thank you. Okay, well, of course, thank you, Prateep, for inviting me and organizing this session. And I can't tell you how pleasing it is as a basic neuroscientist to see how packed this this auditorium is, that there are people who actually care about basic mechanisms and what they might tell us about how this class of substances work. So I started my lab in 1990, before some of you were born, and I've always been interested in using drugs as powerful probes of nervous system function. For a while, I was studying drugs of abuse, such as cocaine and methamphetamine, as again, a powerful way of causing an experience in an experimental animal, and then delving into the brain and seeing what changes occur that account for the behavioral effects. But at least in the early 90s, I didn't think it would be prudent for my career to admit that I had an interest in studying both the classic hallucinogens such as psilocybin or LSD, and never mind a drug such as MDMA, which was getting a lot of bad press, especially in the Bay Area for a variety of reasons. But finally, I came out of the closet about 7 or 8 years ago and published a commentary with my then postdoc, Boris Heifetz, who's now an assistant professor at Stanford and a close collaborator. And we pointed out that studying the actions of MDMA is not, really could provide insight, not only to how MDMA works, but because of its very powerful pro-social effects in human beings, might actually tell us something fundamental about, I think, some of the most important and powerful components of human interaction. And towards the end of my talk, if there's time, I'm going to talk about empathy in mice and how MDMA might actually influence that. Of course, as a psychiatrist who once saw patients, I was also very interested in elucidating the actions of MDMA because, as you all know, it's being tested in a variety of clinical trials, most notably the MAPS trials and using MDMA as an adjunct for psychotherapy. Okay, so Boris was a postdoc in my lab. We teamed up, and he started doing mouse behavioral experiments. And the first thing he had to do was develop a simple behavioral assay for what I'm going to call either social preference or sociability. And the assay he chose to do is actually a fairly standard one. Sorry, I'm having trouble with. Oops, sorry. I can do this. Where is the button? Help me out. No, that's the slides. Did you get it? Which one? The left one? This one. Aha, I did it. Whoops, no. Sorry. Which one? Up here. I'm not going to use the goddamn pointer. All right. So he developed a behavioral assay. He didn't develop it. He used one that's standard in the field, shown in the upper left, which is known as the 3-chamber social preference assay, where you put your mouse in an experimental apparatus. It has 3 rooms. In one room, there's a little cup with a juvenile mouse, and in the other room, there's either an empty cup or a cup with a toy mouse. And you basically ask your subject mouse, which room do you want to hang out in? With the assumption that if it hangs out more around the cup with its little buddy, that's showing social preference or an increase in sociability. So in the upper right in B, what Boris did is a dose response of the effects of MDMA. And what you can see in a dose response, dose-dependent way, MDMA enhanced social preference or sociability in this admittedly very primitive and simple behavioral assay. In C and D, what you'll see is just the time course of the effect. And then in panel E on the lower right, one of my favorite experiments. And by the way, those of you who heard this talk yesterday in Charité, I apologize. It's the same talk. I was too lazy to put together a new talk. But maybe you'll learn something the second time around. So if you look at the bottom right, it's again, one of my favorite experiments. If you look below the graph, you'll see in the second bar, if we gave the MDMA to the mouse that was under the cup, there was a trend towards increasing sociability or social preference. If we gave the MDMA to the mouse that was free to explore, which is the standard way we did this assay, we saw a pretty clear increase in sociability or social preference. And then the last experiment I call the rave experiment, if we gave both mice MDMA, we had the biggest effect, which is actually, it's cute, but it actually may be telling us something about how MDMA actually is working. Okay. So one of the goals of this original project that was published now several years ago was to see if we could mechanistically, either at a molecular level or at a circuit level, dissociate what I'm going to call the pro-social effects from MDMA from what I'm going to define operationally as the abuse liability of MDMA. And what do I mean by that? MDMA is a psychostimulant. It's a part of the same family as methamphetamine and cocaine. I don't think it has nearly the abuse or addictive liability of those drugs, but nevertheless, because it's a psychostimulant, I think it has that potential. So, and I think that's a particularly important point, and Rick Doblin doesn't like it when I talk about this, but the way MDMA is being used in the MAPS trials, I think it is very safe. It's used as an adjunct for psychotherapy. It's only given twice. but I also anticipate, should the FDA in the United States approve the use of MDMA, even with the black box warning on it, that there's going to start being a lot of off-label use of it. And that poses the potential for inappropriate use and perhaps some use that will do some damage. So in mouse work, it turns out that every drug really pretty much ever tested, not only the psychostimulants, but the opiates, nicotine, alcohol, all drugs that have some form of abuse liability have two behavioral effects. One is very simple. They just increase the locomotor activity of your subject mouse. So what's shown on the left is in a dose-dependent way, MDMA at 7.5 milligrams per kilogram did not cause an increase in locomotion, but a higher dose did. Perhaps more pertinent to, in quotes, abuse liability is assaying the rewarding property of a drug, in this case MDMA. And the gold standard for determining the reinforcing or rewarding quality of a drug is doing some form of self-administration under an instrumental conditioning protocol in your mouse. That's kind of a painful thing to do. So a poor man's version of looking at the rewarding quality of any experience, in this case a drug, is what's known as conditioned place preference, where as shown in the diagram, you give MDMA and you limit the mouse to one specific context, in this case, the gray room on the left. On another day, you give it a control substance like saline and you put the mouse in a different room. And then on the test day, you ask the mouse, where do you want to hang out? And you assume if it wants to hang out in the room where it had a drug experience, that indicates the drug is reinforcing or rewarding. And you can see at the lower dose MDMA was not reinforcing, but at the higher dose it was. Okay, so what are the molecular targets of MDMA? We already heard from Dietmar that it's well established that MDMA, its major molecular targets, and certainly not its only ones, but its highest affinity molecular target is the serotonin transporter, which we call SERT. It also interacts with the dopamine transporter, which I'll call DAT. It has been reported to cause the release of oxytocin via an indirect mechanism. And there's a lot of indirect evidence saying increases in oxytocin levels may contribute to some of the behavioral effects of MDMA. We're gonna focus initially on the role of the serotonin transporter and whether that's important for mediating this pro-social effect of MDMA. So the first thing Boris did is a simple behavioral pharmacology experiment. He gave MDMA IP, but before doing that, he gave a drug called citalopram, which is a high affinity binder to the serotonin transporter, which prevents MDMA from binding the serotonin transporter. And you can see that that prevented the pro-social effects of MDMA. He then took a molecular approach using the modern tools of neuroscience, meaning really a genetic approach. We obtained a conditional knockout of the serotonin transporter, which allowed Boris to delete the serotonin transporter only from the dorsal raphe neurons that produce serotonin. And you can see in panel E, that knocking out the serotonin transporter from dorsal raphe serotonin neurons, it's noisy data, but on average blocked the pro-social effects of MDMA, saying its molecular interaction with the serotonin transporter is necessary. Importantly, in those same mice, if we used a higher dose of MDMA and assayed its reinforcing quality in this conditioned place preference assay, that was not affected by knocking out the serotonin transporter. It also wasn't affected by citalopram. So right there, that tells you that the molecular target that is mediating the reinforcing quality of the MDMA experience, it does not require the serotonin transporter. Okay, so where is MDMA acting in the brain? I hope most of you are neuroscientists and know there's something called the classic mesolimbic reward circuitry, which classically consists of dopamine neurons in the ventral tegmental area that send projections to another key component of this reward circuitry called the nucleus accumbens, also known as the ventral striatum. I didn't have time to go through this, but my lab has done a lot of work doing manipulations that suggest that the release of serotonin, specifically in the nucleus accumbens, promotes prosocial behaviors, promotes social reward, promotes sociability. So given that MDMA was interacting with the serotonin transporter, and I'll show you later, causes a massive release of serotonin in the nucleus accumbens, it was an obvious experiment to test whether directly infusing MDMA into the nucleus accumbens, this is old-fashioned behavioral pharmacology, but I still think there's a place for that in modern neuroscience. And what you can see in the purple bar here is that direct infusion of MDMA into the nucleus accumbens had a very robust enhancement of sociability in this 3-chamber assay, saying that its action only in the ventral striatum is at least sufficient for this specific behavioral effect of MDMA. Doesn't mean for many of the other effects, nucleus accumbens is the target. He also did the converse experiment where he gave MDMA IP in the belly of the mouse, and he infused the serotonin transporter antagonist, or the inhibitor, citalopram, directly into the nucleus accumbens. So this is just a test of this hypothesis that MDMA is acting on SERT within the nucleus accumbens. And you can see in the right bar that on average, this manipulation blocked the prosocial effects of MDMA. So we have 2 different behavioral pharmacology experiments suggesting it's MDMA interacting with the serotonin transporter, most likely on the serotonergic inputs in the nucleus accumbens that's required for its prosocial interaction. Importantly, this is another condition-place preference assay, infusing citalopram into the nucleus accumbens to prevent MDMA from interacting with the serotonin transporter. It did not block the condition-place preference caused by MDMA, whereas a non-specific dopamine receptor antagonist infused in the nucleus accumbens did, which is not unexpected. This is just a panel from a paper we published in Neuron recently on a related but different topic, where we used a genetically encoded sensor of serotonin, which we expressed in neurons in the nucleus accumbens, and then we gave the animal MDMA IP into its belly. And all this what this shows you is that, as expected, MDMA does cause a massive release of serotonin in this target structure because it receives a pretty heavy serotonergic innervation. So, of course, we were interested in the potential therapeutic potential of MDMA. I have a specific interest in autism spectrum disorder for a variety of reasons. So we, in subsequent work done by Jessica Walsh, she obtained actually 6 different mouse models of autism spectrum disorder. I'm going to show you data from 3 of them. This is, these first 2 are genetically based models of autism. What's graphed on the upper graphs is a different assay. It's called the juvenile intruder assay. I'm going to go for 25 minutes and leave 5 minutes. I'm just seeing you test it. I know I always go over. I'm well known for going over. But I flew a long way from California. And I am jet lagged. All right, so the top graphs are, it's a very simple assay. It's called the juvenile interaction or intruder assay. You take your subject mouse, you place a little buddy mouse in its cage, you use a juvenile so you reduce the normal aggression between conspecifics of the same sex. And what you can see, if you look at the blue bars, you can see that both in that assay and then the bottom graphs show the assay we've been doing, the 3-chamber social preference assay, that these autism mouse models have a deficit that is largely rescued by giving MDMA. And then this is a 3rd mouse model of autism. This is an environmental model where you give pregnant moms valproic acid, and for reasons I don't understand, the pups show deficits in a variety of behaviors related to autism spectrum disorder. And again, what you can see in the blue bar, in the blue lines, is these mice show a deficit in these sociability assays that's rescued by MDMA. How is MDMA working? It's going to be complicated, as Dietmar beautifully showed you. Serotonin causes the massive release of serotonin. What did I say? MDMA causes the massive release of serotonin. I think there are, 16 or 17 different serotonin receptors in the brain. So it's really hard to figure out which of those receptors the released serotonin caused by MDMA is critical for the behavioral effect you're studying. For a variety of reasons based on previous work, we were specifically interested in a receptor called the serotonin 1B receptor. And what this shows you is that if we infuse a 1B receptor antagonist directly into the nucleus accumbens, we can block the behavioral effects of MDMA. Finally, and this is a plug for colleagues here from MindMed who are pursuing the possibility that different enantiomers of MDMA may have therapeutic benefit. So why is that? The MDMA that hopefully you guys don't buy off the street but get from a reliable source, and more importantly, the MDMA that people are using in clinical trials, in particular MAPS, is a racemic mixture of two different enantiomers. It's RS-MDMA. And if you go into the literature and you look at the affinities of the R versus the S enantiomer, you look at some of the behavioral assays that have been done, it looks like the R enantiomer has a higher affinity for the serotonin transporter and a lower affinity for the dopamine transporter. And based on our hypotheses, that would suggest that the R enantiomer should still have prosocial effects, but might have a reduced abuse liability. And this just shows in a small number of animals, it's only like 5 or, well, it's a very small number, that our MDMA did enhance sociability in this simple assay. So there are actually 2 clinical trials going on based in part on this kind of work. A company called MapLight is pursuing a serotonin 1B receptor agonist in autism, and MindMed is pursuing doing early work to try to get an IND around our MDMA. What does this have to do with empathy? So, as some of you know, MDMA, people in the field, I think, disagree and argue whether MDMA is an empathogen, meaning it actually enhances, in quotes, empathy, however you want to define it, or as you know, God, who coined the term? Was it Shulgin who called it an entactogen? Is that right, Matthias? Oh, it was David Nichols. I'm sorry, it was David Nichols. In part because does it actually influence empathy? Does it actually influence how I interact with the person in my environment? So we decided to see if we could develop behavioral assays of what I'm going to call behavioral antecedents of empathy in mice. And I'm going to tell you very quickly in the last 5 minutes about some assays, a postdoc in my lab who now has her own lab at UC San Diego, which is called the social transfer of pain. And it's actually a fairly simple assay. You take 2 mice, you put 1 mouse in pain by injecting a toxic substance in its hind paw, and then you take another mouse, which we call the bystander or observer mouse, and you just let it hang out with the mouse in pain for an hour. And then that observer mouse will exhibit a bunch of different behaviors that indicate that it is experiencing pain. And one way of showing that on the lower left is doing a mechanical sensitivity assay it's a very standard assay in the pain field using things called von Frey, whatever. Again, what you can see on the lower left is the animal with CFA has a very low mechanical threshold for withdrawing its paw, meaning it's in pain. The purple line shows the bystander mouse. So this is a mouse that has never experienced physical pain, just hung out with another mouse in pain. And you can see it manifests behaviors indicating it's actually in pain, and these other graphs just confirm that, for about 4 to 12 hours. We also developed an assay of sort of the opposite effect, and this is the social transfer of analgesia. So in this assay, both mice are in pain. So both mice have received an injection of a toxic substance in a hind paw. So they're limping around. We then give 1 mouse morphine. So it's analgesic. It's no longer experiencing pain. We take our other mouse who's in pain and just let it hang out with the mouse that is experiencing analgesia. And what you can see is that its pain threshold has increased. Its mechanical sensitivity has decreased. That's the blue lines. So it's pretty wild. But again, this should be intuitively obvious if any of you have kids, for example. If you're a parent and your kid is sick, it makes you feel awful. You want to take that pain away. So I am anthropomorphizing this a little bit, but I would argue that these are behavioral antecedents of what we term empathy. So does MDMA affect this? And then I'll wrap up. So this is the key result. This is hot and not published yet. So this is just the pain threshold, the mechanical threshold in the mouse that's in pain. It has a very low mechanical threshold. Here's the bystander mouse. Here now, we only let the bystander mouse hang out with the mouse in pain for 10 minutes, a much shorter interaction period. And in this, for some reason, they show a little bit of increase in their pain, a decrease in their mechanical threshold that lasts about 1 to 4 hours. If we give the bystander mouse MDMA, we dramatically enhance that social transfer of pain. So that mouse experiencing MDMA is now much more sensitive to the affective state of this mouse in pain. And obviously we did control experiments. MDMA itself does not affect pain thresholds. What is this? I'll skip that. What is this? So this just shows you in a different way that direct infusion of MDMA into the nucleus accumbens enhances the social transfer of pain. So in the middle bars, graphs on these slides where it says saline, there's a little bit of an effect. There's a little bit of a social transfer of pain, but not much. But in the animals that received MDMA infusion into the accumbens, there's a very clear social transfer of pain. And then, I know I'm going quickly, but the take-home message is we did the same experiment with the social transfer of analgesia, and MDMA IP enhanced the social transfer of analgesia. I don't have time to go through the bar graphs, but you can just see the blue bars are much higher than the other bars. And then finally, we again did the experiment of infusing MDMA directly into the nucleus accumbens, and again, that compared to infusion of saline in the accumbens, that enhanced the social transfer of analgesia. So I actually finished more or less on time, which is very unusual for me. So the take-home message is MDMA promotes prosocial behaviors and, in quotes, empathy. Empathy as operationally defined by these primitive social transfer assays in mice, at least in part by causing large increases in serotonin, specifically in this target region of classic reward circuitry. And this requires an interaction with the serotonin transporter. Serotonin 1B receptors in the accumbens are responsible for some of the effects of MDMA. It turns out the serotonin 1B receptor antagonist does not seem to be blocking the social transfer of pain, meaning there's some other serotonin receptors involved in that. And I would argue, and this is a good topic for the panel discussion, that the importance of this kind of preclinical foundational neuroscience work is maybe by understanding how a drug like MDMA works. Not only can we make better versions of MDMA with more of the therapeutic components of its effects and reduce its potential side effects, its potential toxicity. But I also think by using MDMA as probes, we may actually learn something about the neural mechanisms of empathy and perhaps even compassion. And I would argue what is more important in the world today, given the state of our world. So with that, I will end, and I'm happy to answer any questions. And the people who did the work, I just sit in front of my computer, so I don't do any of this. All right, well, 1, 2, sorry. Go ahead. Because it works. Operationally, it actually, we can quantify pain behavior using like these mechanical thresholds. It's funny, so you're asking, why didn't we look at the social transfer of reward? Is that, we're actually doing experiments it's not quite that. I have another postdoc, he's doing experiments. I don't know whether to call it helping behavior, compassion behavior. We're asking whether a mouse will learn to work to give another mouse a reward. So it's not quite answering your question, but and it looks like it's working. And then you can play lots of games with that. One other question.
So I noticed this might not have been statistically significant, but I noticed that there was one bar graph you showed in the beginning that showed that homozygous certain knockout animals under the influence of MDMA had reduced sociability. I wonder if you had any mechanistic insight into why that might be.
That's a great question. I go fast because I don't like people. It's a really, you pay attention to the data. You can work with me. Because a lot of people don't pay attention to the data. So what he's referring to is when we genetically deleted all the serotonin transporters in a homozygous knockout, it looked like MDMA was actually reducing. And the answer is, let's talk offline. I don't have a good answer for that. All right, thank you very much.
Thank you for your talk. Yeah, and already kind of paving the way for the panel discussion. Thanks a lot, Rob. So next up, we'll have Dr. Torben Ott. Torben is a relatively new group leader in Berlin, and I'm happy to welcome you. And Torben will speak about striatal mechanisms for hallucination like perception in rodents. So.
Okay, yeah, thanks a lot, Prateep, for inviting me and putting together the symposium. And as part of our mission to better understand how psychedelics work, we focus on hallucinations and hallucination-like perceptions. And these are processes that we can study both in humans and in rodents. So basically, I want to show you that we can build up an argument that mice can hallucinate, or in a sort of super small version, like a mini hallucination, but I'll walk you through that. So let me see this thing. Aha, good. So in this recent book, Anil Seth has said, and this is quoted now kind of a lot, that we can view perception as a controlled hallucination. And what is meant by that is that we don't perceive our external environment exactly as it is, but that our percept is strongly influenced by our prior expectations. And here's an example that I borrowed from a review from Phil Corlett, where we're looking at a scene here of a man from behind holding a device to the head, which looks something like a hairdryer. Now, this might be a very common scene in the context of, say, a bathroom, and your prior expectation of seeing a hairdryer is fairly reasonably high. It's in accord with the input from the sensory system, and your percept might pan out accordingly. Now imagine a different scene. You're outside in the night, in a big city, in a dark alley, and you see something like this. Now your prior expectation might be quite different. You might not expect to see a hairdryer, right? And you might see a gun, for example. So what I'm arguing here is that we can view hallucinations as an extreme version of this process of integrating sensory information and prior expectations. And if we take this seriously, this means that we can study the neural mechanisms of hallucinogens by studying normal and aberrant perception. And this gives us a way into the neural basis for psychedelics. And even then, even when taking this seriously, it's not that easy. Why? Because perception and hallucination are still subjective. And these are subjective experiences, so how can we access them, especially in animal models? And our focus here is to study the computational processes that describe perception, perceptual inference, and this links candidate neural circuits with both the subjective content of perception and with observable behavior that results from these processes. And then we can see how we can translate now from humans to animals, because we can study the same information processes that give rise to similar analogous behaviors and identify neural circuits, even if we don't have access to the subjective content of perception. So concretely, I'll present a study in which we developed a behavioral task to model what we call hallucination-like perceptions in rodents. And this is to also emphasize that we're talking here about a reductionist, reduced version of hallucinations. We then looked into neural circuit mechanisms, concretely into striatal dopamine. Now, I'd like to highlight Katharina Schmack here. She spearheaded this study. She runs now a lab at the Crick Institute in London, and this work was conducted in Adam Capit's lab in Washington University in St. Louis. Now, again, going back to the idea of perception and Bayesian inference. Here, we will characterize a hallucination as a false percept, a percept in the absence of evidence, perceived with high confidence. The percept looks or feels real in some way, even if you know it isn't real. And we then devised a behavioral task that combines classic ideas from psychophysics with a novel component of a confidence report to study this perceptual process. So here we use mice trained in this task. The mice are walking around in this little behavior chamber, and there is a wide background noise that is played back almost at all times. And then sometimes we embed a faint signal, a frequency stimulus in this background noise. The mice perform a series of decisions. They start what we call a trial, an individual decision process, by poking into the center poke. And here we're looking at a signal trial where now the poking triggers the playback of this faint signal. And then the mice can make one of two choices. It can go left or it can go right. And here right means that it heard a signal, which is the correct answer because this was a signal trial, and this will lead to a water reward. And in the signal detection theory field, this is called a hit trial. Now, if the mouse goes to the left side, that indicates that it did not hear a signal. This is a mistrial, it will not be rewarded. Now we randomly interleave no signal trials. Now here, the contingencies are the opposite. Now here the mouse has to go to the left side, indicating that it did not hear a signal, which is here is correct, which will also lead to a water reward. While if it goes to the right side, indicating that it heard a signal, this is incorrect, will not lead to a water reward, and this is called a false alarm, and these will become particularly important in the subsequent analyses. Now, in the second portion of this task, we measure behaviorally confidence. So to measure this, the idea is here, the trick is that we randomly delay reward delivery for up to 5 seconds. And nothing tells the mouse whether it's going to get a reward or not. It has to rely on its own estimate of being correct. And so in these incorrect trials, false alarm and mistrials, and a subset of correct trials, 5% of probe trials, there will never be a reward. So the mouse commits to a choice, it anticipates a reward, it waits until it has to at some point decide when to stop waiting, leave in order to restart a trial. And this waiting time we consider an investment, and we will then analyze whether this reflects confidence in this task. Then to reiterate, here we operationalize now what we call hallucination-like perception, HALIP, as false alarms. These incorrect choices heard a signal even though there was none. Perceived with high confidence, meaning the mice invested a long amount of time. Now let's first look at the choice behavior of the mice. So here we're looking at the percentage of signal choices as a function of the signal to noise ratio. That's the loudness of the signal that was embedded in the noise. And we see if the signal was loud, the mice always report signal choice, that makes sense. And then if the signal is faint, they sometimes report signal, no signal. And now importantly, here are all the trials, all of the no signal trials. And still in about 20% of cases, the mice report hearing a signal. Now, this idea is not particularly novel, but what has been very difficult is to interpret these error trials. Because there might be a number of reasons for those. It could be just at some point that don't pay attention or they're not motivated, whatever that exactly means. So how can we know whether a false alarm is related to the perceptual system? And here confidence again comes into play, where we now use a model for confidence, where we define confidence as the probability of being correct given the amount of evidence that you have at your disposal, which here is your percept. And this model makes a number of quite specific predictions that we can test in the behavioral data. So this is now a model here where the amount of confidence going higher, predicts the accuracy in the task, of course, because that's how it's defined. But also confidence increases with the signal to noise ratio in correct trials, but decreases in error trials, or in other words, the same sensory stimulus. For the same sensory stimulus, confidence tells you whether we're correct or incorrect. Also, if we look at high confidence versus low confidence trials, we see that in high confidence trials, we have a steeper psychometric curve, again showing us that these trials tend to be more correct. Now, when we look at the data of an example mouse here, we see that the data follow these quite specific predictions, and this allows us to argue that time investment serves as an appropriate behavioral readout for confidence. So then we have both now false alarms and high confidence. So let's look at common manipulations that have been used also in the human literature to induce these simple kinds of hallucinations or hallucination-like percepts. And first here, we're looking at strong signal expectations. So we're inducing a strong expectation of a signal, which in humans is associated with perceiving, having a wrong percept. And so now we have stretches of trials where we show 70% of signals versus 30% of signals, and it shifts the psychometric curve exactly as we would expect. Namely, we see an increase in false alarms when we have a high signal proportion, but also an increase in the amount of time the animals invest, meaning an increase in their confidence report. Then second, we also injected a cohort of mice with ketamine. And again, we see a small leftward shift and, or at least an increase in these false alarms and amount of time investment here in this task. Now, we also ran a human version of this task where we used the exact same stimulus, the same white noise, the same signal, and similar signal to noise ratios. In this human version, we had online participants wearing headphones, and they indicated whether they heard a signal or not with a button press, and reported their confidence on a scale. In humans, it's a bit easier, so we don't need to do this trick with the time investment to get a readout of their confidence. Again, here's a psychometric curve of human participants. Very high, always correct, you know, when the signal noise ratio is high. Again, we have around, in humans, around 10 to 12% typically of false alarms. And then we added psychotic phenotyping here with questionnaires to these human participants. And specifically, we used the CAP score to quantify hallucination self-report. So these include questions like, do you hear sometimes voices? And even though this was a general cohort, we see a quite broad distribution of these CAP scores. So we asked whether this would relate or be correlated with the performance in this task, namely with having false alarms with high confidence. So here we see the CAP score related to false alarms and the CAP score related to the confidence in false alarms. And here we saw a somewhat small, but still a correlation in this exploratory study. What was even more interesting that we also added other dimensions here, depression, anxiety, and so on. And this was the only one that showed this kind of correlation. So, before we go to the second part, to briefly summarize, we have this behavioral task that we can run in animals and in humans. And we have done at least a couple of validations for this task, where we now argue that we can quantify hallucination-like perception. Now, to start digging into the neural circuits, there has been a long-standing hypothesis also for, in the schizophrenia research, that striatal dopamine could play a role in psychosis. So, for example, there's increased turnover of dopamine in the striatum. And this was one of the motivations here to look first into striatal dopamine. So we here measured the relative concentrations of dopamine in the striatum using dopamine sensors similar to the serotonin sensors that we've seen before. So these are modified dopamine receptors that become fluorescent when dopamine binds to them, and it allows to measure their concentration in vivo during the behavior. We focused on two subregions of the striatum, the ventral striatum overlapping with the nucleus accumbens and classically associated with reward processing, and the tail of the striatum, which at least some animal studies have shown it could play a role more in sensory processing or novelty. And so here we implant a fiber and we express a sensor and we then measure fluorescence. Now here we see this fluorescence signal, the proxy for dopamine concentrations across time, and here aligned to the stimulus onset in the task. And we see somewhat similar-ish responses, meaning there is a higher, the amount of fluorescence here after stimulus onset is correlated with the signal-to-noise ratio. But what we also observed is that even before the stimulus comes up, there was a fair amount of trial-by-trial variability in the amount of fluorescence. And this we see here. So this is now the baseline fluorescence just before the stimulus comes up. And here we see a difference between the false alarm fluorescence levels and the correct rejection fluorescence levels. So these are all no signal trials, but we see a higher fluorescence whenever later on the mouse would report a signal in both areas. And even more so, we also saw a correlation between the strength of the signal and how often the mice would respond with a high time investment false alarm. Now we saw this in both of these areas, and to further interpret what these signals could mean, we have to briefly take a step back and look at a computational model. Like this is a fairly standard model that would explain this perceptual inference process. Briefly, the idea is that your percept, as I said in the very beginning, is a combination of both a prior expectation, that's often called a prior, here denoted with mu pi, and the sensory evidence that you have, sometimes called likelihood, if we're using full distributions. These are then combined using Bayes' theorem to form the posterior distribution, and your percept is sampled from this posterior. Now, your percept then determines the choice. You heard something or didn't hear something for a detection task, and also the amount of confidence that you have. And then eventually you receive some feedback, whether you made a correct choice or not. And this feedback then is scaled by the amount of confidence to form a prediction error, and this prediction error updates the signal prior, the amount of expectation that you have. So this model, naturally, because it's designed for this kind of perceptual inference process, reproduces the psychometric curve and these confidence-specific signatures. But now, importantly, there's two ways in the model to obtain something that looks like a hallucination like percept, or specifically an increase in these false alarms. And one way is by having, prior to hearing a stimulus, having a higher prior reward expectation. And that corresponds in the model to a high absolute value of your prior mu pi. This would lead to false alarms, more false alarms, but also to more misses, which actually was not something we saw in the behavioral data. The other way is having an increased signal expectation. This increases your false alarms, but also your hit trials, which is more in line with what we saw in the behavioral data. Now, when we revisit our dopamine data from the two strial subregions, what we saw is that the predictions from the model for reward expectation versus signal expectations, there are like two key predictions here. One is that the amount of reward expectation, the absolute value of the prior, is correlated with the probability of being correct, but not with the probability of making a signal choice. And vice versa for signal expectation, where it's a bit more intuitive, perhaps, that the amount of signal expectation correlates with you making a signal choice. And when we look at the data, what we saw is that in ventral striatum, the data, at least qualitatively, resembled more the reward expectation process versus the tail of the striatum, fluorescence, dopamine levels, resembled more the signal expectation data. So this means, at least here in this study, the tail of the striatum is a particularly strong candidate to signal the prior of a signal. Now, in the final set of experiments, we increased dopamine concentrations in the tail of the striatum using optogenetics, expressing light-sensitive channels in dopamine neurons, dopamine-releasing neurons, and this increased both the false alarm rate and the time investment. This effect could be rescued with haloperidol, and both for the false alarm rate and the time investment rate. And this effect actually has been recently reproduced by a group at the University of Chicago, just published a month ago, which for us is great because, this is one study and not that many animals. So it looks like this dopamine-dependent effect in the striatum is something that could play a big role in these kinds of behaviors. So to conclude, at the end of my talk, we saw that we have this behavioral task to quantify hallucination-like perceptions as a potential model for hallucinations. We can run this in humans and animals. It seems striatal dopamine plays an important role in these hallucination-like percepts. This data is most in line with dopamine in the tail of the striatum signaling the amount of signal expectation. So a little mock circuit, which is mostly hypothesis at this stage, could look like that here in the tail of the striatum, increased dopamine somehow increases whatever signals come in that would convey the expectation. Now, ongoing work in the lab here. So I started my lab last year in Berlin. So unfortunately, just short of being able to share data, I think hopefully next time or so, we are looking at whether we can expand this model into serotonergic hallucinogens such as psychedelics, and then specifically look at how serotonin and serotonin receptors in different brain areas could be related to these hallucination-like percepts in rodents. And with that, I want to thank my lab here in Berlin. There's two people here working on these projects, Eric and Daria. If you want to learn more about these behaviors. And again, thanks to my collaborators, Katarina and Adam. And thank you guys again for inviting me and being here.
So we have time for 2 questions from the audience. Yeah.
Yeah, so Matthew also started with Emil Seth, right? So we're using they're similar frameworks for perception, for sure. And it would be very interesting to apply his theory about dendritic coupling in this context, for sure. There is, I think, a vast array now of experiments that we can and want to do. One of the first ones, I think, would be looking into a number of different areas across the cortical layers and just identifying which areas are even good candidates here, right? And I do think this experiment is something that we eventually should do. Realistically, I think it's going to take time, and we haven't done it yet.
Pathways involved in hallucinations. That is not really what we are more talking about with the psychedelics, for example. Do you think it's going to go also into that pathway in terms of the neurocircuits?
Yeah, so this study was motivated also from exploring new models for psychosis. And here, dopamine has been implicated for a long, long time. Just early antipsychotics acting on the dopamine system, and so on and so forth. Likely, I think there will be multiple circuits involved. So there will be multiple ways of pushing around the perceptual system, and multiple ways of inducing something like hallucinations or full-blown hallucinations. And there are convergence points. I mean, for example, in the animal literature. It is fairly well established that ketamine increases striatal dopamine levels, just to name one of them. And then when you think about the circuits, and for example, you combine sensory information with prior expectations, this is a process that likely also happens in many places. When you think about hierarchical loops in cortex and all of these ideas in predictive processing, and that implies that there are multiple locations or multiple circuits where you could induce potentially different kinds of hallucinations, who knows? Yeah, so there's many ways I think how these could converge.
I have one question for you, Tom. So I'm really curious, one day if we could test this Carhart-Harris hypothesis of rebus, because this theory has been put out there, relaxed belief on the psychedelics, but I think there is a complete lack of mechanistic understanding of such theories which come from human studies.
Yeah, please, anyone correct me also if I'm wrong, that there is basically no evidence for the rebus model, but it is here at this stage, it's a very interesting theory, of course, and it lives in a similar framework. So these Bayesian inference perceptual models, they come out of the even more general models about predictive processing, or all the way up to, for instance, free energy principle. And I think what we have to do is we have to fill them with concrete specific situations, specific kinds of behavioral tests, specific kinds of perceptual processes, and that are sort of special cases of these bigger frameworks. And currently, our data indicates more that dopamine is here correlated with an increase, like more dopamine leads to hallucinations because of an increase in your signal prior, which sounds sort of like the opposite of the relaxed expectations, which is a reduce prior in the rebus model. But that's not necessarily yet a contradiction because, but that becomes a bit technical possibly, but because he's talking about these landscapes of the different priors that you have, and that the switching between those within this landscape is easier or happens. You need less energy to switch between those different states if you're under psychedelics, right? And here we're looking at a fairly simple situation, right, where we have one signal. So that's one of the reasons why I think at the moment we cannot fully argue for one or the other, and they might not be mutually exclusive in the end.
Thank you very much, Torben, once again. So we are in the middle of this symposium, and it's time now to go to more human side of things. So with this, I invite Professor Kim Kuypers from Maastricht, and she will talk about microdosing psychedelics. Where are we, and where to go from here? Welcome.
Thank you so much, Prateep, for the invitation and the introduction. Indeed, we're now moving to the human model, leaving the mouse model, the animal model. Still waiting to get my slides here. So this is the beautiful building where I work in the Netherlands, in Maastricht. And as I said, I'm going to talk about microdosing psychedelics. I will start off with a definition so that we are all on the same page. What is microdosing? It is taking 1/10 to 1/20 of a regular dose. Question is, what is a regular dose? You can base that off research, doses that are used in research. So that would be between 10 and 20 micrograms of LSD, between 1 and 3 or even 5 milligrams of psilocybin or another psychedelic. People are also microdosing with ayahuasca, for example. You take the low dose repeatedly for a period, and it is with the intention to improve certain aspects of life without disrupting any other processes. And it's intended to have, you don't want to have the hallucinogenic effects. So how do you do that? If you want to use LSD, you can buy a blotter and clip a piece off and dilute it in water and then take it. Or you can also, let's see whether I can manage this pointer. You can also buy it online, the truffles, in these packages or like this. And then it's the question, how do you take it? And one schedule is this. It's the Fetterman schedule. You take it on one day. It's the day that you experience the acute effects. You leave on the second day, you experience the subacute effects. And then on the third day, you don't feel anything anymore. That is the hypothesis. And then you proceed on taking another dose. Why the schedule? Fetterman introduced it as a research protocol to investigate for yourself what microdosing does to you. But I looked on the internet, and there are also several other research or other microdosing protocols, and we also learned that from our survey research, where people state that they come up with their own schedule. But I thought this was very interesting because it's running before science. They say how to choose your microdosing protocol. It starts with, are you a beginner? And then you can follow this. Yes, I'm a beginner. And then the question is, do you suffer from a psychiatric condition? If it's yes, then they state use it every other day. If you don't have a medical condition, so you can use the Federman protocol, the one I suggested. If you're not a beginner, then it's the question what your main goal is. Is it to do one of these things? Yes, then use the Stamets protocol that is stated here. Otherwise, use the Federman protocol or an intuitive protocol or 2 fixed days. So what is the Stamets protocol about? It's taking 4 days in a row and then leaving 3 days without dosing. And you also stack it. You use something else together with it. You can combine it with lion's mane or niacin. Niacin, which is used as active, as placebo in psilocybin studies. Just kidding. So my title was, where are we and where are we going to? I want to first start by telling you where we are in Maastricht. And I think our journey started, 6 years ago, when we started off with, we wanted to do a microdosing study, and microdosing is, like I told you, giving repeated doses to people. But then we had the question, what is a microdose? We do not exactly know what it is. So we said we will start off with a dose finding study, a controlled lab study. But in parallel, we also started survey studies. And the first survey study was about, we asked people, why do you microdose and do you use it as self-medication? What came out of this study was that people have different motivations to microdose. It's to enhance concentration, creativity, productivity, but also to help out with mental health conditions. And in this survey, we also showed that we asked them, when you microdose, can you compare the efficacy with when you use conventional medication or a regular psychedelic dose? And we showed that people with ADHD find it more efficient or better to treat their condition, to take microdosing compared to conventional medication. So that put us on the track of ADHD. And we're now also doing a controlled lab study together with Matthias Lichty's group in Basel. So we're doing a controlled study in ADHD patients. What did we do more? So we did the dose finding study, and we showed that we gave people 0 micrograms of LSD, 5, 10, and 20. And we showed that with 20, some people really experience things, so that's not non-hallucinogenic anymore. But they also experienced positive effects, increased mood, positive mood, but sometimes also increased anxiety, increased attention, which is also very good in the context of ADHD. So then we continued with the repeated dosing study that we have now concluded. We gave participants 4 doses with, I think, 2 days in between, 15 micrograms of LSD. I'm not going to discuss these data today. We're still analyzing them. We also conducted the EEG, and I can tell you we saw something in the EEG, so that's good. And we also saw it 4 days after they stopped microdosing. And then, like I told you, we have this, then now I clicked the wrong button. I wanted to show you this. No, never mind. So what I'm going to do now is focus on our, I called it a survey study, but I will explain what it means, a survey study. I will focus on this one, and then shortly on this one. So, this survey study, what does it entail? We designed a prospective naturalistic study, and this is the advertisement. It's in Dutch. I knew it. You probably cannot read it, but this is the way we attracted people. So we asked people, do you think you have ADHD? Are you diagnosed, or do you have symptoms? And do you have the intention to start microdosing with psychedelics? And then they could enter the study. And this was the study design. So we had a baseline measure before they started microdosing. We assessed them at 2 weeks and at 4 weeks in the microdosing process. What did we assess? Multiple things. We assessed emotion regulation and empathy. And oh yeah, there should be something else, it doesn't matter. We also assessed mindfulness and personality. I will not, oh yeah. And then we assessed ADHD symptoms, well-being, and time perception. So you can go and have a look at the poster of Eileen Hyen. It's a PhD student who's working on this project, and she presents these data on a poster. I will not discuss the mindfulness and personality data, but I can tell you that after 4 weeks of microdosing, they had changes in their trait mindfulness, and also their personality. So for example, here, neuroticism decreased. So we're going to focus on this part. So here you can see the flow chart of the participants, and here you can see also that it was a very difficult process, like it's always when you do naturalistic studies. You start off with a bunch of interested, highly motivated people. And then you end up with, so we started off with 365, I think, 56, and we ended with 47. Why is that? Yeah, because people lose interest or because of other reasons. We also had to include some of the people here because they had incomplete responses at baseline or for other reasons. What we did, as I told you, not everybody had a diagnosis of ADHD, and that was really the intention because we're targeting adults, not everybody is diagnosed, and people do experience symptoms. So that's why we used the CARS, the Connors Adult ADHD Rating Scale, to define their symptoms. And you have a cutoff of 65, which states if you are above this cutoff or on this cutoff, you have elevated symptoms of ADHD. So here I show you the group. This is at baseline, at 2 weeks, and at 4 weeks. You see that it were mostly females. Yeah, it's half-half almost. Most of the people came from Europe. They had a high education level. They did mostly computer or office work. And then here you can see how many people had a diagnosis. This is not solely ADHD. This is shown here. So it was a group that you do not see, I think, in controlled clinical trials, because there you really go for the people who have ADHD, but that's not the way it is. People have comorbidities, as you can see here. People had depression, anxiety disorders, PTSD, a lot. We also asked them which psychedelic they used. We used a diary to assess that. Not everybody filled out a diary, but those who did, here you can see that most of them used psilocybin, either mushrooms or truffles, followed by novel lysergium. So the novel, I cannot pronounce it. And then followed by LSD and ayahuasca. That was only one person who used ayahuasca. I think very interesting here, also something that you don't have in a clinical trial, is some people switched from the novel substances to psilocybin, or from LSD to psilocybin, or from psilocybin to LSD. So that was every time one person. We don't have the reason. It can be that they just were not able to acquire it. I think I'm looking at Eileen. We don't have the reasons. No, we don't. So let's go to the first results. So we used the CARS, as I said, to assess ADHD symptoms. It are 30 symptoms, and an example is shown here. I interrupt when others are talking, and then I think, oh, should I do that myself? Not at all. They could rate that on a scale from, I don't know, 0 to 5 or something. Not at all, and very much, very frequently. And this are the things, or this is on the y-axis. You can see the total amount of symptoms represented here, and here you can see the assessment points, baseline, 2 weeks after microdosing, and 4 weeks. And what we show is that they were, and I have to do this. So this, the orange line is the 65 cutoff line, so elevated ADHD symptoms. And this would be then, between those two blue bars, is that you have probably ADHD when you use this survey to assess it. So you see that our sample was well above the 65 cutoff. And after microdosing, they decreased and they went under the cutoff. What is also interesting, I think, because you can say, oh, it's a naturalistic study, you did not include a control group. That is totally true, I know that. But what I do like about our data is that we also had, so in this group, we split it up. You had people using medications alongside their microdosing. And you had people who only microdosed. And for those people who combined the microdosing and the conventional medication, you see that they also decreased, but it went slower, the process. And this was not attributable to the severity of the ADHD. We also asked them about their well-being. We tested that with the WHO 5 well-being index, and this contains of 5 simple items shown here. And they have to say all of the time or at no time. And then you just do the sum score. It's actually from 0 to 100. It's not shown here on the graph. And also here you have a threshold where you say if you're below 50, you really have poor mental well-being or well-being. Here you can see that our sample was really not doing very good, but after microdosing, they increased to, I think it's called then moderate well-being, or in a better range. And here you can also see that these groups, so medication use and microdosing, they differ slightly. Then what we also did is we are also interested in behavior, and it's difficult when doing a naturalistic study, but we gave them a computer task. We focused on time perception because people with ADHD have problems with time perception, but also because in another trial, an effect of LSD on time perception was shown. So we asked them to listen to several tones they had several durations. So they were presented with the first tone of 300 hertz. They had to listen to it. And then they heard a second tone of 440 hertz, and they had to reproduce it. So they had to push a space bar and then, just leave it. Don't push it anymore if they thought that this was the interval. The reason why this is a different hertz is that you cannot simply reproduce it from your auditory memory. What is the dependent variable? You do this. Ooh, that's a bit too much. You subtract their reproduction from the original tone and you divide it by the original tone duration. This is what we showed. Actually, I can be very short on this. We didn't show much. These are the separate intervals, the separate durations. And when you zoom in on one of the durations, you see that there is an effect, and this is the effect. There was an overestimation of this interval only for individuals who used a conventional ADHD medication and microdosing after 2 and 4 weeks. But since it is only on this interval, and you also have to know that there were only 2 trials per tone, we also want to see this reproduced in a more controlled trial, so we're hesitant to draw conclusions from this. So the summary of this part is that in this study we showed improvement in ADHD symptoms after 2 weeks of microdosing and then also after 4 weeks of microdosing, and that there seemed to be a delay in the decrease in ADHD symptoms when people combined their conventional medication with microdosing. What is interesting, we okay, we don't have a control group, but we can compare to norm scores or other studies. And we showed, so the change we showed was also shown in studies that used mindfulness-based cognitive therapy or people who were under treatment with methylphenidate. And something interesting that I will come back to in one of my last slides is that 20% of the sample did not show improvements in ADHD symptoms. So there are people that do not respond. Then the well-being, I showed you increased well-being after microdosing, and these changes in well-being or improvements were related to a decrease in ADHD symptoms. We have to say that we did the study in the Corona period, in the COVID period, and we also compared our scores to the norm scores of a very large study that was conducted in that period too. And they are similar, so similar low well-being. Yeah, and then I have to look at this after two. Yeah. So this shows that our scores are in line with this sample of normal people who were tested in the Corona period. This I already addressed. We do not show a lot on the time perception task, and what I also said, there has to be caution because the sample that showed a difference was a very small sample. I think that there were 9 participants or a bit more in this conventional medication combined to microdosing group. So I totally acknowledge that there are limitations to this study, but there are also strengths. We use validated questionnaires, and like I said, you have norm scores to compare our data set with. You have survey studies asking people, hey, how do you feel, or how do you feel when microdosing? What we did is a step further. You ask people before they start to microdose and then follow them up in time, so you can do some causal inferences. And there is, what I do like is the ecological validity. People choose their dose, they choose the substance they want to microdose with, they choose their scheme, they can do the test wherever they want, whenever they want. The limitations are that I showed you that in one of the first slides, we have a large dropout. We don't know why people dropped out. You cannot ask if people do not respond anymore. We don't have a control group, but we're now working on that. We're asking people to take part in this study for a control group. And we also know that it is a self-selected group. People really had the intention to microdose. But I was thinking about that. So people who engage in lab research also have their motivations to engage in lab research. So in that sense, yeah, it is a limitation, but other studies also have their limitations. So like I said, this is a work in process. We're running a controlled study together with Matthias Lichty Group. It's sponsored by MindMed. I hope that we can conclude it this year. I'm looking at Matthias. He's nodding, yes. So hopefully we will be able to present the results of that study soon. So let's zoom out. Talked a lot about what's happening in Maastricht, but things are happening in the world too. What we see is that there are some controlled studies. Oh yeah, I had to change that. It has increased. I think 8 single dose studies, 4 repeated dose studies, and you can see then that the dosing, like I said in the beginning, people take doses according to schedule. Here you see that in some studies they receive 2 weeks of microdosing, 3 weeks, 6 weeks, and then the doses range. Not all studies use the same LSD, some use base, some use start rate. So it's good if you're working in this microdose range that you have that on your mind. When reading these studies, 5 micrograms of base is, you need a bit more to convert it to tartrate. Psilocybin studies, I'm not sure anymore. I don't think that there are yet really controlled studies. Most of them do it like the self-blinding study by Imperial College, or they ask people to use mushrooms or truffles and then conduct the tasks online or come to the lab. Some studies, what I really like is that they ask for, that they have a sample and that they do then the analysis so that they know how many psilocin or psilocybin was in there. And here you see the details of that. Here it is important that the storing conditions will also influence the content of your psilocybin. What we see is when they give repeated doses or when they take repeated doses, it are 2 doses per week in general. A summary of those, and it's very quick, and it's very short. So what we see is that you have self ratings, you have computer tasks, you even have studies that include imaging, EEG or functional imaging. Some studies show that mood states, positive mood states, negative mood states are increased. Also, some participants show increased attention, creativity. It's a more difficult one. I think one psilocybin study has shown that creativity can be enhanced, but some studies just don't show anything. We showed that people have an increased pain tolerance. So when doing the cold pressure test, they can keep their hand longer in water, cold water. There was a study by the Chicago lab showing changes in brain connectivity, which was positively correlated with changes in mood states. You see a change in oscillations, theta waves, which is theta waves linked to learning and memory. There's one beautiful study by Madsen by the Copenhagen lab that shows that even the tiniest dose of psilocybin also shows receptor binding. They did a PET study. And there is recently a study, it's not yet really published, but it's a pre-published thing. They show that the night after microdosing, the sleep duration is increased. That's also a trajectory we would like to follow to investigate the effects of microdosing on sleep. Then what is shown too, that it's overall well tolerated. People show slightly elevated blood pressure. And what we also showed is when you look at markers, we showed increased BDNF levels after 5 and 20 micrograms of LSD up to, for the 20 micrograms, we showed it up to 6 hours after intake. What you have to remind is that these are all acute effects and mostly single dose studies, mostly LSD studies and healthy volunteers. What we need to know is, what I will also show you in the last slide, is you need to know whether these effects are persistent and whether it helps in patient studies or patients. Oh yeah, I included these. I also like to look for case studies, and I found these, found 2 case studies where people self-administered psilocybin or another I think the other case is also psilocybin. And it helped in the treatment-resistant depression. It was a psychiatrist who assessed with the MADRS the scores, and it really decreased. I think he had a number of rounds with microdosing. Cannot remember the details anymore, but look into it. It's only 1 paper. It's super interesting. And here also another person who had Lyme disease, but then also suffered from depression and anxiety, and then one day also decided to microdose, and this helped a lot with his depression. These are just case studies, but I think there is value in it. It tells you something that it can help people, but it also links to, oh yeah, one more slide, and then I will show you to what it links. The question is, will it be over-the-counter medicine? Because you can say, oh, non-hallucinogenic, people can take it on their own, ADHD, why not? I doubt that. I would like to have a different approach. And what you also see on the internet is when people now sell microdosing kits, they sell a complete package. This is what they do. They give you coaching, you do journaling, you are really engaged in some kind of therapy. And I do think that is good. When you go to the poster of Eileen, you will see that microdosing also does something with emotion regulation. And people might need assistance when emotional processes are changing too. Here you see a quote of somebody who said, having microdosed often before the last 7 years, I can say that the monitoring, group communication, and journaling has a much higher impact than just using it intuitively or even with a strict protocol. I was skeptical of the calibration process, but it did make a difference. What they do is they let people go through a calibration process where they test different doses of psilocybin, and then they can make up for themselves, does it work for me or not, and then move on to the next dose or not. And this is another quote stating, my intention to heal has been accomplished now, which has enabled me to formulate a new intention for going deeper into the process. I also subtly transformed in a pleasant way. All in all, this program has exceeded my expectations for what I can achieve. You can also think, oh, this is promo talk on such a website, can be, but I do think that we have to keep on testing the value of microdosing. And here, yeah, I circled this because intention is also very, important. So, but this also links to frequently mentioned issues. I'm looking at somebody of people who are not that fond of microdosing. So what is mentioned? Yeah, you know, people are breaking blind, and it's due to their breaking blind that they have these positive effects. You can see that in the field, you can see that in the lab, people do sense bodily sensations. And this might cause people to have, if you already have certain expectancies and you break blind, you might steer the results into a certain direction. Another problem is the motivation. Why do people engage in microdosing research, in naturalistic studies? That's also what I also told you. I think the perfect solution, I don't have the perfect solution, but I do think that we have to include, we already do this, including treatment guesses. What we did not do is justification. So ask people, in the example I gave you, we did this dose finding study. It was a within subject study, so people received on 75%, 3 of the 4 times they received LSD. If you have to guess then when you receive LSD, you have a higher chance of guessing correctly. So you need to ask for a justification. Why did you say that you think you had LSD? And then they might say, yeah, well, I felt this or this. And I think you can use that then in your analysis in some way. What I do like is another procedure used by the Chicago lab. They tell you can either get an hallucinogen, a stimulant, a cannabinoid, or a sedative. And with this approach, here you can see with the 6.5 micrograms of LSD, they had no correct guesses. With the 13, they had 2 out of 20 correct. With 26, they had 6 out of 20 correct, and placebo, 14 out of 20 correct. It's also not the ideal solution, but it is a solution, and I can also think of other ways to deceive participants, perhaps. And of course, you deblind them, you debrief them then afterwards. I would like to end with this. I already said in the beginning, this is something that lies close to my heart. I really want to look into this more personal factors that determine your effects of microdosing. What should we look into? The age, biological sex, weight, dosing scheme. Why age and biological sex? Because age, because we know that 5-HT2A receptor binding changes with age. Biological sex, because we know that females in research, in LSD research, has shown responses that are a bit delayed compared to men. Oh, there is something missing here. Yeah, what was also an interesting one is how you metabolize the substance. If you're a fast metabolizer, I know that there are not a lot of people, but they might, the substance might not reach the system. Should we also investigate whether there is tolerance after microdosing? Is it true that people have to uptitrate? We have to know about the working mechanisms so that we know which indications could benefit from microdosing. We have to look into the therapy format, and we also have to look into safety. If you're going to look into the literature, you see that some people are concerned about this. But in these articles, you also see that more research is needed, and it's not very sure that this will happen with microdosing. These are the references I've used, and I want to thank you, and I also want to thank the whole team that worked on these projects. Thank you.
Any questions? One question for Kim? Yeah. 1 second, we get you the microphone.
Yes, thank you.
Thanks so much for your talk. I had a question about the splitting into the medicated and unmedicated groups. I was wondering if you also looked at that in terms of the diagnosed and undiagnosed, because I'm just anticipating a criticism, particularly from like psychiatrists, of like, oh, if they're medicated, they have to have a diagnosis, and therefore, is it just that it's two subgroups that are rather than a pharmacological difference, and maybe the people who are undiagnosed watch a bunch of TikToks and they're suggestible and they're more prone to placebo or something like that?
Yeah, I think we had more people who were microdosed than people, and I'm looking at Eileen, that were not diagnosed, right? We did not look into, no. So the people were grouped, everybody was tested on the cars, also the people who were diagnosed. And I hear your criticism, but yeah.
My anticipated criticism, not my criticism.
No, but it is a valid point, but I also think, and we hear that more, that not all people who are diagnosed are correctly diagnosed. So I think it's a fair process to have them all do the CARs. But I also hear you that these diagnosed people were the ones who were taking the ADHD medication. Yeah, I cannot give you a good answer to that. We did correct for comorbidity, and that did not change the data. And we also corrected for something else. Medication use, that did not change the data. Yeah.
Thank you.
Thank you.
Thanks once again, Kim. So before the panel, our last speaker is Professor David Olson from UC Davis. Welcome. David, can you hear us? Can you hear us?
Yes, I can hear.
Yeah,
yes, I can hear you. Just give me 1 second to share my screen.
Sure. The talk is titled Mechanisms of Psychedelic-Induced Neuroplasticity. Thanks, David.
Does this look fine to everybody?
Yep.
Good, great. All right, let's get started. So thank you for the invitation. I'm sorry I couldn't be there in person. So my name is David Olson. I'm the director of the Institute for Psychedelics and Neurotherapeutics at UC Davis. Before we get going, I also have to tell you that in addition to my academic appointments, I'm also a co-founder and the chief innovation officer of Delix Therapeutics. But everything I'm going to tell you about today was work that was done in my academic laboratory. So our group is really interested in finding new treatments for so-called stress-related neuropsychiatric diseases. These are conditions like depression, post-traumatic stress disorder, and substance use disorder. And a hallmark of all of these illnesses is the atrophy of key brain regions, in particular, the prefrontal cortex. And the prefrontal cortex is really critical because it controls motivation, fear, and reward. And a hypothesis in the field has been that if you could identify chemical stimuli that can induce the growth of cortical neurons, such as the growth of new dendrites, or if you zoom in on one of these dendrites, you'll see the growth of these dendritic spines. If you can promote that type of growth, you might be able to achieve a few different really interesting results. Number one, you might have a rapid acting response. Number two, the response might be sustained over a long period of time. And number three, the treatment might actually work across indications because the PFC is such a critical a critical hub. And so when we're looking for these types of chemical stimuli, one of the first molecules, probably the best known molecule that has this ability is this molecule here on the left, this dissociative anesthetic called ketamine. And so here I'm showing you some cultured cortical neurons that were treated with ketamine, and you can see that they have increased number of these dendritic branches, and these little green dots are representing synapses. So they also have an increased number of synapses. So ketamine promotes both neuritogenesis and synaptogenesis. And now I'm showing you some in vivo work that was performed by the late Ron Duman and the late George Agajanian. What they were able to show is that chronic stress can lead to that cortical atrophy that I told you about, the loss of those dendritic spines in the prefrontal cortex, and that really manifests as synaptic deficits that you can measure with, traditional techniques like electrophysiology. But what is really remarkable is that a single dose of ketamine within 24 hours can rapidly regrow those lost dendritic spines in the cortex and reestablish synaptic connectivity. Now, I think ketamine is super exciting for the field because it's pushing us in a completely new direction, a direction that is more in line with a healing-based approach, trying to repair these damaged neural circuits. But the issue with ketamine, there are many. One of the issues is really its kind of lack of circuit level selectivity. The primary target of ketamine is the NMDA receptor, which is found broadly across the entire brain. And so when we got into this area, we were thinking very deeply about what receptors might be selectively expressed in the specific neurons and brain regions that we really care about for treating these stress-related neuropsychiatric diseases. And that really led us to study the serotonin or 5-HT2A receptor. Here I'm showing you the expression patterns of the 5-HT2A receptor across the rodent brain. And you can see it's heavily expressed in these layer 5 pyramidal neurons of the cortex, with more expression in the anterior parts of the brain, less expression as you move towards the back of the brain, and much, much lower expression in structures like the hippocampus or the striatum. So one of the first experiments that we did was simply to take cultured cortical neurons, treat them with psychedelics, and look at neuronal morphology. And you can see that compared to the vehicle-treated neurons in the middle, the neurons treated with the psychedelics, DMT and LSD, have substantially more complex dendritic arbors. And if you do some super resolution microscopy and zoom in on one of these dendrites, you'll also see an increase in the number of dendritic spines and these filopodia-like structures. Now, there's a number of ways to quantify changes in cortical neuron structural plasticity. I'm going to briefly tell you about one way that we use fairly regularly. It's a very simple method. It's called Scholl analysis. The idea is you just draw these concentric circles of increasing radii starting at the soma of the neuron, and then you simply count the number of times the dendritic branch crosses one of those circles. And you can produce these really, really beautiful Scholl plots like the one here on the right, where on the x-axis are the radii of those concentric circles, and on the y-axis is the number of crossings. But typically the way we like to represent the data is simply looking at the maximum number of crossings for a particular drug. And so for the vehicle control, it would be about 7 here. For the DMT, it would be about 11. So looking at these NMAX plots, what we can see is that psychedelics from 3 distinct structural classes, ergolines like LSD, tryptamines like DMT, and amphetamines like DOI, they're all very good at promoting cortical neuron growth. Now, all of these early experiments that we were doing, we were treating cultured cortical neurons for 72 hours. And we know that in vivo, that's really not what happens. In vivo, you give a psychedelic drug, it rapidly gets into the brain, and then it's rapidly cleared. But there's very long-lasting effects. And so we were very interested to see what is the shortest stimulation period that we could use that would produce long-lasting changes in neuronal structure. So instead of stimulating for 3 days, we started pulsing with for 24 hours or an hour or maybe just 15 minutes. And then we take the drug away and then give the neurons time to grow and look for changes in morphology. And what we find is that even with these really short stimulation periods, just 15 minutes or an hour, you can get very profound increases in cortical neuron growth. But the trick is you have to give the neurons time to grow. If you just stimulate for an hour, fix and stain, you see absolutely no change in morphology. But if you stimulate for an hour, give the neurons a couple of days to grow, then you can see pretty large differences. So to demonstrate this effect in vivo, we relied on using a psychedelic that is rapidly cleared from the body, dimethyltryptamine. The half-life of DMT in a rodent is only about 10 minutes. And so for this experiment, we gave a single IP injection of DMT to rats. We then waited 24 hours, a time period when all of the drug has been cleared from the body. And we then sacrificed the animals, look at neuronal structure via Golgi staining, and function via electrophysiology. And what we find is really interesting. We find that even after the drug has been cleared from the body, we get this long-lasting increase in dendritic spine density, and that's accompanied by functional effects, including increases in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. Now, since we published this work way back in 2018, it has been replicated and extended by a number of groups across the world. But I want to focus on one study in particular because I think it's really, really interesting. This was work done by Alex Kwan's group back when he was at Yale. He has since moved to Cornell. But Alex was really interested in seeing just how long do these effects last for. So he gave a single dose of psilocybin to male and female mice, and he finds that psilocybin induces increased density of cortical of spines in the prefrontal cortex that lasts for over a month, which is just really remarkable to me. So at this point, we're very interested in trying to understand the mechanism by which psychedelics promote this cortical neuron growth phenotype. And so we started with just some selective pharmacology. So the very first thing we did is we tried to block the serotonin 2A receptor because it's well known that the serotonin 2A receptor mediates the hallucinogenic effects of psychedelics. And if you give a 5-HT2A antagonist to people, you completely block the subjective effects of those drugs. And what we find is that the 2A antagonist can completely block the effects of psychedelics from the 3 different structural classes. And then the next thing we did is we tried to look at some downstream kinases. These are 2 kinases that were implicated in the plasticity promoting effects of ketamine. The first is called TrkB, which is the high affinity receptor for brain-derived neurotrophic factor. And again, we find that if we block TrkB, psychedelics are no longer able to promote cortical neuron growth. And the same is true of this downstream kinase, mTOR. mTOR is really important for producing all of the proteins necessary for plasticity, all of the structural proteins that allow neurons to change their shape, and all of the ion channels that allow neurons to change their synaptic weights. So at this point, this was decent evidence, but we realized that the antagonist that we were using to block the serotonin 2A receptor is not perfectly selective. It hits a couple of other receptors. So to be absolutely certain that the 5-HT2A receptor was the target, we relied on genetic models. And so here we used animals that have had the 5-HT2A receptor genetically deleted. And we can see that in wild type animals, the psychedelic 5-methoxy-DMT produces a very robust effect on cortical spine growth, but the effect is completely gone in the 5-HT2A knockout animals. We're not the only group to observe this. Javier Gonzalez-Maiso's lab has seen a very similar result with a different psychedelic, an amphetamine type psychedelic called DOI. Now, we also looked at neuronal function. And so here again, you can see that in the wild type animals, you get this increase in the frequency and amplitude of these spontaneous excitatory postsynaptic currents. But in the 5-HT2A knockout animals, the effect is completely gone. So now we wanted to see if the 5-HT2A receptor might be involved in the behavioral effects of psychedelics. And so for this, we relied on a couple of different assays. I'm going to show you first this one, which is a modified version of the forced swim test. And we've modified this in such a way that traditional antidepressants like SSRIs are not very effective, but these next generation antidepressants like ketamine tend to be effective. And so the way we do this is we run a forced swim test on the first day to just do 2 things: establish that all groups have exactly the same level of immobility, and also provide some additional stress to the animal. And then we give our dose of compound. We do a behavioral test, immediately after, just to ensure that we've engaged the target appropriately. And then 24 hours later, after the drugs have been cleared from the body, we then do our assessment of efficacy. And what you can see is that when you give 5-methoxy-DMT on its own, you get this decrease in immobility, this antidepressant-like behavioral response. But if you use the 5-HT2 antagonist ketanserin, you can completely block this effect. Now, the forced swim test is probably a little more relevant to motivated behavior. So we wanted to look at an assay that is more related to anhedonia. And for that, we relied on the sucrose preference test. So mice, much like myself, will prefer a sugary drink over just plain old water. And you can use this preference as a measure of anhedonia. And so the way we do this is on the first day, we give them a choice between sucrose and water. And you can see that they all prefer the bottle of sucrose. We then can induce an anhedonia-like phenotype by giving some kind of chronic stress. And in this case, we're giving chronic administration of corticosterone. And then following 10 days of core administration, we do another sucrose preference test. And you can see that in both the wild type animals and in the 5-HT2A knockout animals, there is this drastic reduction in their preference for the sucrose bottle. On day 13, we give a single injection of psilocybin, and then 24 hours later, we measure sucrose preference again. And you can see that in the wild type animal, psilocybin causes a very nice rescue of this sucrose preference, but you don't see that same effect in the 5-HT2A knockout animals. So at this point in time, we're feeling pretty confident that the 5-HT2A receptor mediates the effects of psychedelics on structural cortical neuroplasticity, and also maybe some of the antidepressant behavioral responses. But there was a piece of data that had us just scratching our heads for the longest time, and it's this one. And it was the fact that this molecule right here, serotonin, could not promote cortical neuron growth. And that was really surprising to us because I just spent the last 10 minutes telling you that the serotonin 2A receptor was responsible for psychedelic-induced structural neuroplasticity. And so it didn't really make a ton of sense until we got this particular structure-activity relationship study finished. We started making some small modifications to serotonin-like molecules over here on the left. And we noticed that as you started to add methyl groups, going from one methyl group to two methyl groups, you saw a much bigger effect on cortical neuron growth. And you can see that here in these images, but it's very easy to see this with these show plots. So going from this molecule here, tryptamine, to N-methyltryptamine to dimethyltryptamine, you see very big increases in cortical neuron structural plasticity. So then what we did was we tried to correlate the activity of the 5-HT2A receptor when it was heterologously expressed in a non-neuronal cell like a HEC cell with cortical neuron growth. And we saw absolutely no correlation at all. If anything, we saw a slight negative correlation. But when we tried to correlate the lipophilicity of the drugs, how greasy are the drugs with their ability to promote cortical neuron growth, we saw a very nice positive correlation where these very polar drugs like serotonin and tryptamine could not promote cortical neuron growth, but these non-polar drugs like these psychedelics are very good at promoting cortical neuron growth. And that led us to the hypothesis that maybe the reason that serotonin cannot do the same thing as something like DMT is because maybe it can't actually access the target. The 5-HT2A receptor is a G protein coupled receptor, and canonically, many GPCRs exist on the plasma membrane, and they're meant to interact with molecules at the plasma membrane and translate that into intracellular signaling cascades. But in this case, we thought maybe the 5-HT2A receptor was on the inside of the cell. And if that was the case, serotonin via a zwitterionic compound is too polar to passively cross the nonpolar membrane and access the target. So the first thing that we did is we simply wanted to look for, where is it located in a neuron? But before we did that, we wanted to see where is it located in just a HEC cell. And so here I'm showing you the expression patterns of the 5-HT2A receptor, a different GPCR, the beta-2 adrenergic receptor, and then this is just the CFP control. You can see that the CFP goes everywhere in the cell, but the two GPCRs are localized where you would expect them, kind of on the plasma membrane with some intracellular as well. This white line is just a plasma membrane marker. But when you look in neurons, in cortical neurons in particular, you see something very different. You see that for the 5-HT2A receptor, most everything is localized intracellularly, but the beta-2 adrenergic receptor still has lots of receptors on the cell surface. And so these are live cell imaging. We also wanted to do this doing fixed cell imaging so that we could try to localize exactly which subcellular compartment the 5-HT2A receptor was found in. And I should note that, this was mainly a confirmation study here. We're not the first to show that the 5-HT2A receptor is intracellular in pyramidal neurons in the cortex. This has been shown by many, many other groups over the course of, I don't know, maybe 20 years or something, but no one really understood what was the functional consequence of this intracellular localization. So when we tried to do our subcellular localization analysis, we found something really interesting. And that is that the 5-HT2A receptor co-localized extremely well with markers of the Golgi. If you look in a hex cell, you don't see the same thing. So here is 5-HT2A receptor expression. You can see there's a bunch of 2A that is not localized with the Golgi marker. So it's very different in cortical neurons versus hex cells. So next, to try to understand this a little better, we developed some new molecular tools based on these membrane permeable compounds here in green. So we've got the membrane permeable agonist DMT and psilocin, and the membrane permeable antagonist ketanserin. And we just converted them to their membrane impermeable versions in purple by methylating DMT or putting a phosphate group on psilocybin or methylating ketanserin. So all the compounds in purple are permanently charged, and so they cannot passively cross a nonpolar lipid bilayer. So then what we did is we ran the assay under two different conditions. One is just normal, plain old cortical neurons. The other one is with a little bit of electroporation. And with electroporation, you can apply a voltage to transiently open pores in the neurons. And what that does is it allows the membrane impermeable compounds to cross the membrane, the pores close, and then the impermeable compounds get stuck on the inside of the cell. And when we do that, we see something really interesting. Without electroporation, we get the effects that you would expect. DMT and psilocin, the two membrane permeable agonists, are very good at promoting cortical neuron growth, whereas the impermeable agonists, TMT and psilocybin, cannot. Similarly, the membrane permeable antagonist, ketanserin, can block the effects of DMT, but the membrane impermeable antagonist, methylated ketanserin, cannot block the effects of DMT. But when you electroporate the cells, you get the opposite. So now we can see that TMT and psilocybin can promote the growth of cortical neurons, and methylated ketanserin can block the effects of DMT. So these are just some neuritogenesis studies. We also did this with spinogenesis, and we saw the exact same effect. The membrane permeable compounds promoting growth, the membrane impermeable compounds not promoting growth. So at this point, we reason that if it really is all about the location of the receptor, serotonin should be able to do the same thing that a psychedelic can do if we can just get it to the target. And so normally the serotonin transporter is expressed on presynaptic serotonergic neurons. It's not expressed in postsynaptic pyramidal neurons of the cortex. And so for this experiment, what we did is we sparsely transfected the transporter so that we could import serotonin into a select number of neurons. And I love this experiment because within the same culture, you have some neurons that can import serotonin and some neurons that cannot. So looking here at the vehicle side, we've got two neurons that are sitting side by side. The one on the left expresses the transporter, the one on the right does not. Similarly over here, again, two neurons side by side, the one on the right expresses the transporter, the one on the left does not. And so then we can treat the cultures with the various compounds, and we find that in both genotypes, the vehicle doesn't really do anything. And DMT promotes the growth of both types of neurons, both genotypes, presumably because DMT can just passively cross the membrane, so it doesn't require the transporter. But what's really interesting is that serotonin can promote the growth of the neurons that can import serotonin, but it cannot promote the growth of the neurons that cannot. And so this next study was, a partial replication of the last one. So you can see here the same effect, serotonin promoting the growth in SERT-positive neurons. But then we wanted to assess the effects of the 5-HT2A receptor and the transporter. So the other thing that we did is we tried to block the effects of the 5-HT2 antagonist ketanserin. And you can see that the effects are completely blocked. But then we also use citalopram to block the transporter itself. And again, it seems that serotonin requires the transporter. So this study really indicates that serotonin requires the transporter to get inside the cell, and then it requires the 5-HT2A receptor to produce the growth effects. Now, if you just change serotonin for a membrane permeable compound like DMT, you see something very different. Again, DMT can promote the growth of both genotypes. Ketanserin can block the effects of DMT in both genotypes, but citalopram cannot block the effects, again, because DMT does not require import through the transporter. So these were all neuritogenesis studies. We ran the exact same studies in spinogenesis, and here are very similar results. Again, no effects of the vehicle, DMT promoting spinogenesis in both genotypes, but serotonin only promoting spinogenesis in SERT-positive neurons. So to test this hypothesis in vivo, we relied on a genetic strategy. So we used a virus to express the serotonin transporter on the postsynaptic side on those cortical neurons. And then after 3 weeks, to allow the virus to express, we then gave a single injection of parachloroamphetamine, a selective serotonin releasing agent. And then 24 hours later, we come back and we look at dendritic spine density. And what we find is that only in the neurons capable of importing the monoamine do we see this increase in cortical spine density. Now, if increases in cortical spine density are important for the antidepressant-like behavioral effects, we would expect to see something very similar using this paradigm. And so what we did is, again, express the serotonin transporter using a virus. We then ran a novelty-induced locomotion assay just to make sure there was no differences between the genotypes, and there wasn't. We then do our forced swim test, the pretest, again, showing there's no difference between the two genotypes at baseline. We then gave a single dose of PCA, came back 24 hours later, and what we find is that we get this antidepressant-like effect only in the animals that have the serotonin transporter on the postsynaptic side and can import the released serotonin. So in summary, what I tried to tell you about today is the fact that psychedelics can promote cortical neuron structural neuroplasticity through the activation of a particular intracellular population of 5-HT2A receptors, really highlighting the role of location bias in their signaling. And then finally, I'd like to thank these groups for funding. And most importantly, I'd like to thank all the incredible students that I get to work with every day. I've highlighted the students or I've highlighted the names of some past students whose work I highlighted today. But you can see all the people in white. These are current lab members who, unfortunately, I didn't have a chance to talk about a lot of their work, but maybe sometime in the future. And with that, I'd be happy to answer any questions.
Maybe?
Hello, thank you for your talk. I don't know, I've been heard?
I can hear you.
Okay, I couldn't see you, so I didn't know. Okay. Yes, well, your paper is incredible. It's been a hot topic this last year for many discussions. And well, before reading your work, I have read the work of Dr. González Maeso and also Dr. David Nichols, which in the early 2000s already had this question of why some 5-HT2A agonists do elicit these psychedelic responses, and while others do not, like serotonin itself. And their work was more, well, they discovered like that bias agonism of the 5-HT2A receptors in the cytoplasmic membrane was very important. And actually there was a different engaging of signaling molecules depending on if on the nature of the agonist of this of 5-HT2A receptors in extracellular receptors. Yeah, you've got even they showed that some signaling cascades that are related to plasticity were engaged, like the MAP kinase cascade or the EGR transcription factors. So, I mean, I was wondering, how would you integrate all this previous research on the field with your team's discoveries? Like, is it all about these intracellular receptors, or maybe there are like different mechanisms that are acting all together, and these are like different parts of the same story? So
yeah, so first thing I want to say is that we were just talking about one very specific population of 5-HT2A receptors, just the ones that are in the cortical neurons of the PFC. There's 5-HT2A receptors in lots of other brain regions, and their signaling mechanisms could be completely different. The other thing is that the 5-HT2A receptor is well known to exhibit functional selectivity. What the consequence of that functional selectivity is still very much an open question. A lot of those functional selectivity studies were done in things like HEX cells, where the signaling might be different than in various neuronal populations. And that might be different depending on the brain region. And the other question, so I guess the question is G protein versus beta arrestin. When people talk about functional selectivity, that's typically what they're talking about, although there's more aspects to functional selectivity. I will say that if you just look at the biased signaling of psychedelics in a HEX cell, and then you try to figure out which ones are hallucinogenic or not, there's really not much of a correlation. There are G protein biased ligands that are hallucinogenic. There are beta arrestin biased ligands that are hallucinogenic. And I think the field is now coming to the point where it's generally accepted that G protein and beta arrestin differences, it might not play much of a role. And it seems that G protein signaling is probably the bigger, more important effect. But I want you pointed out the fact that Dave Nicholas said this, but Dave Nicholas also, which is really intriguing, has some old work, and it was really what inspired a lot of this, that he demonstrated that the lipophilicity of psychedelic drugs tracked really, really nicely with their hallucinogenic potency in people. And I think he was just hypothesizing that had to do with their ability to get into the brain, but it might also be their ability to just cross membranes, period, and access this intracellular target. And so molecules like LSD are very interesting because their pKa's are very tightly controlled, such that a large proportion of the molecule exists as both the protonated and the deprotonated state at physiological pH. And that allows them to very easily cross membranes.
Thanks, David. Did you want to add a comment to this question? You had your hands up, or do you have a question?
So can you hear me now? So
yes.
I have a question and a comment. So the question is actually related to the previous one, but specifically, so now you have these pyramidal cells that either express serotonin transporter or not. So what is the difference in the signaling of those receptors that are at the surface or inside? I mean, why would they be different? And that's the question. And the comment, okay, the comment is that there's been work by Patricia Gaspar a long time ago, but largely ignored. She showed that during the early development, many pyramidal neurons actually express serotonin transporter during the time of growth. So I'm just wondering whether this could be related to your work, so that serotonin might be actually acting as a sort of more effectively.
Yeah, no, it's a really good point. And so, in early development, there's a ton of plasticity that's going on. And if there is transporter expressed on the cortical neurons at that time, then serotonin could be a very effective ligand for that system and might be responsible for the early structural plasticity that you see. But in the cortical cultures that we do, we don't see any expression of the transporter unless we overexpress it. At least it's not expressed at high enough levels that it could really import a lot of serotonin. And then the first question was about the signaling. The answer to that is it's unknown. We don't really know if these different populations of receptors signal differently, or if it's just about which ligands are able to access them.
Yeah, but I mean, if they produce different effects, they should actually have a different signaling, you would expect, wouldn't you?
Well, it could also be just the quantity. It could be that, there's a small number on the cell surface, so you don't elicit a full response. But there's a lot more of them intracellular. It's possible.
Thanks very much. I think that concludes all the speakers. Thank you very much. I would like to invite everyone. So for the panel discussion, I would like to invite our speakers up here. And David, you can still hear us, right? Can you
Yes.
see us as well?
Yes.
Yeah, perfect. Okay. So Kim, Rob, Dietmar, and Torben, please come up front. And there are many chairs in front, so now whoever has been sitting on the floor can grab chairs. So we are waiting for Dietmar? Yeah. We can start a little bit with a general question. So, am I missing Dietmar? Yeah, okay. No, probably is. So, the general question, I think this is for all the speakers in the panel. What do you think is the grand challenge or the main hurdles in combining or bridging the gap between basic science and clinical research in psychedelic neuroscience? It's for all of you in the panel. So now Dietmar is here. So the question is, what is the grand challenges or the difficult hurdles in bridging the gap between basic research and clinical research in psychedelic neuroscience?
Well,
This is like to all speakers.
Start. I mean, and I always feel like I'm going to state the obvious. One challenge is those of us who are working in, especially rodent models, is we need to develop more sophisticated behavioral assays that hopefully, and it's very challenging, are going to be more predictive of the behavioral effects and perhaps even the subjective effects in human beings. I mean, I'm looking, David, how's your baby? Good. David presented, nice work on the force swim test, but everybody in the field knows that's a primitive assay for trying to assess motivation. So there are better ways. I mean, I'm just using that as an example of trying to assess motivation and decreases in motivation in stressed animals and improvements in motivation. There are better ways of testing hedonia or anhedonia. I mean, the challenge is they're more labor intensive. So, I mean, and again, I feel like I'm stating the obvious. I think David's work, among others, is really valuable because, everybody in this audience knows the huge question in the topic of the therapeutic potential of psychedelics is, do you need the subjective experience or not? And David is appropriately pushing the idea with his brilliant chemistry that perhaps the subjective experience is not necessary, that you can create drugs that don't cause a head twitch assay in mice, so I think the types of tools David and others are creating are going to be incredibly valuable, and it's one of the reasons I started my talk. I like drugs as probes of brain function because you can give them to our rats and mice and you can give them to human beings and you can go back and forth. What else can we do to build better bridges? I mean, obviously, circuit analyses, doing the best we can, trying to figure out in a more sophisticated way what functional connectivity changes that are, using fMRI measurements in human beings. What does that actually mean? And how do we mimic that and model that in animals? And I can go, sorry, I'm going to shut up in a second. I've thought a lot about this, and there's a lot of, I think, bridges that can be built that are not specific for psychedelic science. They're going to be useful for broad areas of psychiatric neuroscience, but I'm going to let somebody else talk.
Dietmar wanted to comment?
Could I just follow up on what Rob said real quickly before we move on? I just want to say I agree completely with what Rob said, but I also want to emphasize that rodents are not people. They will never be people. And so to develop, the behavioral tests are going to be challenging. It's always going to be challenging to get really good behavioral tests that mimic a human condition. When I was a postdoc, I was trying to develop a genetic mouse model of schizophrenia, which is probably the dumbest thing ever because they don't have the brain region that is mainly impacted in human. It's a uniquely human condition. So what I think could be really impactful for the field is the development of translatable biomarkers. If we understand the circuitry better in rodents, then maybe we can just go directly and look at circuitry in humans, whether that's some kind of imaging method, EEG, who knows? But I think that will help the translation.
Yeah, can I, before you, just to add something, or something I also thought about. I've done a lot of MDMA research, and one thing I always saw in the literature is the translatability of the dose. And that is my big question. How do you determine the dose? Because when you showed your data on MDMA, you said 7.5 milligrams per kilogram. I was already calculating. Wow, massive dose. So why is that then a low dose, and why do these animals not feel it? I also know that there is a difference in metabolism, and it also is a difference whether you use a mice or a rat. So, yeah. So how do you determine it?
I think Gita Knudsen would say that you should do it with receptor occupancy using PET ligands, right?
So I would say there's not a single cure, single answer to this question. So first, let me say I'm a big promoter of basic research. I think also without any thought on translation. I think this is important, that people also have the freedom to just do basic research, and often then things pop up. And I think there it becomes important. So one thing I would like to bring up here, I was asked to coordinate a big grant, a collaborative research grant. This is how funding in Germany often is organized. And this was on translational neurosciences. And now 15 years later, there are a few things where we have improved. So really talking to each other sounds so simple. I mean, there's not one way to do it, but we put in, my very simple idea in the beginning was trigger some interest for strong natural science people for medicine. And the grant I'm coordinating, they are really very strong biophysicists, physicists, biologists, guys who have discovered optogenetics and so on. So to interest them for medical issues. One good example was we hired somebody from genetics. His main model was Drosophila in the beginning. Now, 15 years later, he has a clinical study on autophagy and aging, which was kind of impressive. And then what we had, a couple of things. What is the main issue actually that we are not able to translate? Is it the model? There are many different animal models, but for example, the robustness sometimes of data. So this is one guy, a clinical guy I work with a lot from neurology. He said, well, let's have a reproduction really of the study. And it turned out, I think NIH, there were 20 studies, looked good, and then they were reproduced, and one was able to be reproduced. Not that they fake data, it's sometimes not so easy to exactly in terms of methods. So this is his idea, to make things really clear, value and open science and blah, blah. This is his thing. Then we had one module, it's called Spark. I think it was initiated actually at the Stanford University. It's a kind of a small mentoring program with little money for basic researchers. It's a kind of a, not mentoring for junior people, it's really mentoring kind of, and translates this and bring also sometimes industry in, because they want to reduce risk and they give all the stuff then back to academia. And then the last thing is, clinical research units, right, where you can translate. So there's not a single one answer to this question. And specifically for psychedelics, I would say in terms of also of guidelines, right? I mean, for 50 years, we were not allowed to do a study. I think episode is the first one we have in Germany. I think Switzerland and Netherlands are perhaps a bit further ahead, but for 50 years. And now I would say within a year or two, I think the psychedelic field actually in the clinical research is moving relatively fast, I have to say.
You can also briefly comment. I also think there's not one solution, but to go a little further on the behavior point that has been raised, rodents are not humans, but I do think we can do much more than what we are currently doing. Like the tools have been laid out for many years, say in psychophysics and in others, and I think we can do more in having translational studies that look at comparable computational processes, like the notion of computational validity, for example, that some people are discussing in the field of computational psychiatry, where we're looking at similar cognitive processes. We can back translate, we can maybe not make some of the behaviors more, the human behaviors more animal. It's hard to translate in animals, but we could also more often perhaps go the other way around and look at behavioral studies in humans and how we can design some that might resemble more things that we see in animals. And along these lines, I think there's a lot still that we can improve here.
I mean, I think I really, to follow up on that, I was thinking what Dietmar's saying. The other, I mean, and this is the importance of various academic centers starting psychedelic science centers. David has one at UC Davis, obviously there's the Hopkins Center, is bringing preclinical neuroscientists and people studying human subject, clinical neuroscientists, together in the same physical location with shared funding will make an enormous difference. I mean, historically, at least in the United States, it's been virtually impossible for individuals to write a grant where there's two, let's call it rodent researchers and two human subjects researchers. That's slowly changing. And again, I think that's what's the excitement about psychedelic science, is an opportunity to actually build those bridges where, I mean, to me, the most important thing is for people like me to talk to people like you, to hear what's going on in the human subjects world, to pay attention to the challenges people studying human beings face, and vice versa. I think that could go a long way to building bridges, just pragmatic things like that. So it always comes down to money, funding.
Addition, I mean, we are talking about basic researchers, clinicians, scientists, or clinical research. Here is an additional factor which I found interesting. And this I thought about when Rob yesterday gave his talk. So MDMA, is there a long-lasting effect or not? LSD, is there a long-lasting effect? Most likely. But MDMA, for example, it's interesting. It's an adjunct, right? So we need also to bring in people from psychology. So psychotherapy, that's also an interesting, very important variable, I think. This we have to include there. And I think there's also no standard there yet, right? I mean, how to do this, when exactly and how. Mm-hmm.
What do you guys think?
I have a comment. What about employing the public? Like, I would argue that, I don't have an estimate, but more than 99% of the psychedelics are being used not in clinical trials or treatment facilities, but out there in festivals, parties, raves, and there's a huge amount of people who are would be very excited to participate, not just participate, but contribute to the field. What about getting the scientists out there to the? There are some projects I heard about with Burning Man, Zendo project, but I think there's not enough of those.
No, I totally agree. But we tried it a couple of years ago. We went to a psychedelic festival in the Netherlands, and it was very difficult to get our hands on participants or to get good data out of these people because they combine a lot of things. It's a really rich field, but it's very difficult. And we were not able. We had, I think, 100 participants or something, but we were not able to get something out of the data. So another way, I also work together with Quantified Citizen. This is an app you can install for people who microdose or not, and you can engage in such studies. But then also you will have selection bias. There's somebody who wants to add something.
Yeah, I mean, I'm not an expert there, but I mean, it's an important, valid point. I have a really nice roundtable discussion with a couple of clinical colleagues, and there was one idea that we approach one of these more serious, let's call it this way, retreat centers, for example, in the Netherlands, and to see whether we can do something there. It's work in progress. It's not my expertise, but we have beside the clinicians also a psychologist, and we just started to make contacts there. Another thing I found interesting actually is I met somebody who's a very experienced psychiatrist, and he said, well, now he sees patients and they have this experience. And for them, it's really not easy to talk to them. I mean, they don't have the experience. And they also, there's sometimes, then I would say, these patients come in and they have another knowledge or more knowledge, so to say. This I found also an interesting aspect, actually. Yeah. And this guy also then, he wanted to have also such an experience to be able more to talk to the patients. Yeah.
Retreat studies, or we go to retreat studies. We do not run them. Yeah, but But the thing there is also that they actually only include people who are healthy normally, and you also want to have the broader spectrum. But also there, it's difficult to. What we like to do is follow them up, and like I showed you, you have high dropout rates also there with. Field work, you want to have a control group, also very difficult. We now have a control group, but it is difficult. We're trying, and we want to get the data out of the people who really do it, but difficult.
I have a question for David. I have a question for David first. When Twitter was still Twitter, so I follow your lab tweet. This is quite a while ago. I read that you have one of your new psychoplastogens in trial. Would you like to update us a little bit about this?
Yes, I can say some things. I can't say everything, I think. So this is a non-hallucinogenic psychoplastogen. It went into phase 1 clinical trials. The trials have not finished, and so the data has not read out yet, but so far the drug has been very well tolerated. That's about all I can say.
Or if it's X, yeah. Thanks. So, questions? Yeah. One second. Can we have the microphone here, please?
I had the wonderful experience of attending the MAPS conference in Denver and was astonished to meet all sorts of people who told me they were clinicians and that they were doing treatment with psychedelics. And since I'm very professional conference oriented, I was like flabbergasted. What are your training to be doing clinical treatment of patients with psychedelics? Nobody had an answer, and now I see people are offering trainings in America for clinicians to do psychedelic treatment. And what are they doing? And do you have any way of assessing those people to tell them what they should be doing? I think you need to find an answer to that, because if that doesn't work and people have bad experiences, one good case in the newspapers in America and this whole field will go backwards. It's very dangerous, and that's what I'm concerned with.
Yeah, you have very valid points, but I also think that there are many underground therapists who have done this work for years and years, and we should get data from them. I have also been approached by these people, and then it's also the thing, like, yeah, they don't have ethics approval to collect the data, so you cannot do anything with the data, but still we need to find a bridge there to find out what they do, to have the good practices, and then base the training on that. Yeah.
It's very frustrating.
Anyone else from the audience? Yeah. Can we? Thanks.
So my question is a little bit more about the science. I feel like one of the biggest issues facing psychedelic neuroscience is the replicability. And that, most likely has to do with a lot of the factors. The rodent strain that you use, the behavioral assays often have very small signals. But I think that's exacerbated by the fact that we're using, first of all, psychedelics, and there's multiple psychedelics that we can use. So as leaders in the field, how do you guys think that we're gonna come to sort of a consensus on these really big ideas that a lot of labs are having a really hard time replicating?
It's an incredibly important question. Issues of replicability in science have been around for 40 years. It's not a crisis, it's always been an issue. I'm gonna go back to something I said. It comes back to funders. And David probably knows about this. So I'm doing an experiment in the Bay Area with a philanthropist has given me money that I have distributed to 6 different labs in the Bay Area, 5, I think, actually, two at UC Berkeley, 2 at UCSF. These are universities in the Bay Area, 1 at Stanford. And we are all giving our mice psilocybin, same dose, same source of psilocybin, same strain of mice. And we are running a battery of very simple behavioral assays. We just got together for a 5-hour meeting for the first time to compare data to see. And I think that is the only way, that kind of organized scientific enterprise, because if the funders don't give you money to do replication studies, it's going to be the Wild West. It's going to be tons of findings that that are probably going to be wrong, not because anybody is fraudulent, but because of all these variables that can't be controlled. So that's my personal feeling, is how we can start addressing it. And I think it's particularly important in this field, because these, all it's going to take is, what you said, is some bad stuff to happen, and the field's going to go back to the way it was, at least in the United States, after, in whenever it happened in 73 or something. But there may be other answers to that question.
Yeah, this is also what I meant with robustness of data. That this one colleague of mine is arguing that this is the main bottleneck for the translation. So what are we personally doing about this? So starting right at the beginning of the chain, introducing electronic lab book. That was the first thing. Sounds so trivial, yeah? I had a really high-tech guy in the lab, and he said, well, you guys are still using these old books. You have all these fancy machines to write in. This was by law. It took me 2.5 years with lawyers that we are allowed to use electronic lab books. Sounds simple, but it's very transparent. Everyone can look into it. Also, later on, when you publish something, that's the first thing. Then the data acquisition. You need a data manager, and it has to be open, value and open science. So we have specifically somebody hired as a project manager for value and open science. He also looks at all our methods papers. Now, the methods, that this is as transparent as possible and that people can reproduce this. And I can keep going. I mean, this is a very important issue, and then journals are looking more and more into this, and funding agencies. And this is sometimes a bit, I had this discussion actually this morning. I mean, if you put pressure, scientists, yeah, you have to do this, of course, they will do it. But I think having intrinsic motivation is even better. If you put out the data there and you have clever people looking into this data, you sometimes find interesting stuff. I mean, this is perhaps a better motivation for some of the people. Yeah.
Briefly pick up on your last point, like data sharing, and could, one of the things, not by far not the only thing, but one of the things that could help because of exactly what you had said, and alongside, but that's easier said than done. It's really, really challenging. I mean, I started a lab last year, and I was thinking about this a lot, and there were lots of discussions. The Bernstein Network in Berlin, for example, they have data sharing platforms, for example. You need a lot of resources. And I realize now, well, where is the money supposed to come from? I really struggle, like getting any money to have resources for that, for example. And so unfortunately, I don't really have a good solution here. There are some ideas, they are a little bit in place. It seems there is some movement. Having open science is one example, but it's a really big problem.
You want to add a comment to that? You have your hands up.
Yeah, I want to emphasize that I think that a lot of people who have never done behavioral neuroscience, I think they look at it and say it's simple. It is really, really hard because there are so many variables that you just can't even imagine. Even if you have like a perfect protocol, everyone follows it to a T. Maybe there was, I don't know, some construction in the hall or a loud noise, happen, when you were running an experiment. There's so many factors that go into it that make it variable. And so even if you can control for everything possible, the behavior is still going to have pretty big error bars from person to person, lab to lab, even within the same lab. We found there's differences sometimes between whether a man or a woman runs the experiment for just small little things like that. So what we've been doing instead, we still have to use behavior because that's kind of the standard for the field. We've been trying to focus more on direct measurements of circuit activation. So looking at things like immediate early gene changes in neuronal structure, change in electrophysiology. What we have found is that seems to be much more replicable from person to person, lab to lab than the behavior. And we hope that maybe we can find something that would be good enough to translate into a measurement that we could do in a human.
Like an online question that you want to...
Yeah, we have a question from our online audience. So Josh Hartman asked, do you have an opinion on Moliners et al. Publication suggesting that psychedelics promote plasticity via direct binding to TRKBs? Have you replicated their findings?
Is there in the audience, so...
Why did you ask me? I wrote about that in the paper.
Who's going to take the microphone? You want to answer that one?
Yeah, I'm the wrong person to comment on that. Maybe David. I mean, I would like to hear David's comment on that.
I can't tell. Is that Ira? Is he there?
Yeah.
Yeah, that's you.
I was the one that was asking your question before.
So, I mean, I think it's a really intriguing finding. Of course, it got to be replicated. But I think, who knows? We'll see.
I think it's time to end the panel. We guess we are at the end of the day and pretty tired and looking forward to the rest of the evening on a more social note. So thank you again, everyone. Kim, Rob, Torben, Dietmar, and David for tuning in from early morning. So I hope you had some coffee or get ready for the day. Thank you again. Thank you, the audience, for taking part.